Ok. Bueno, ahora vamos a movernos un poco de los rotores al sustrato y vamos a hablar de remodeling. Y creo que hubo una pregunta, espero que la persona que hizo la pregunta esté ahora aquí para poderla contestar. ¿Cuál es el papel de remodeling? ¿Qué es remodeling? No, realmente todos hablamos de remodeling, pero no concretamente. Dobry ya nos dijo un poco de lo que pasa en remodeling, a pasos muy generales. I am going to speak in English, and I'm going to try to give you what my idea of remodeling is. First of all, I'm going to tell you that we simply do not know what the mechanisms of atrial fibrillation in the human heart are. Particularly, we are absolutely in unknown territory when thinking about persistent atrial fibrillation, let alone permanent atrial fibrillation. Now, even the classifications are really very arbitrary because many patients who supposedly are in persistent atrial fibrillation all of a sudden can return to paroxysmal atrial fibrillation, and these things can happen in many, many patients. So the mechanisms are really very poorly understood. So we really need to understand the mechanisms. Unfortunately, there is a need to understand mechanism for a number of reasons. First, there's a rapidly increasing number of patients who are 60 years or older and who are probably going to be 9% of those patients are probably going to develop atrial fibrillation at one time or another. There is a failure of antiarrhythmic drug therapy to maintain sinus rhythm and to reduce mortality. And we know that there is very significant evidence that the major cause of stroke, embolic stroke, is atrial fibrillation. 30% of patients with stroke seem to have atrial fibrillation at one time or another. And there's a rapidly increasing application of radiofrequency ablation, and in many cases we have no evidence-based medicine to support radiofrequency. And many people are doing ablations without really knowing what the consequences are. And finally, there's an expanding impact of healthcare costs both in Europe, the United States, and all over the world. In the United States, there are 2.5 to 3 million people with atrial fibrillation. In Europe, there are 5 million people with atrial fibrillation. And that impinges significantly in ability to work, healthcare costs, and so on. So the cost is tremendous for society. I already talked about the vicious circle of atrial fibrillation. There are triggers, there's substrate, there are rotors, there's high excitation. Atrial fibrillation, when it's maintained for long periods of time, leads to remodeling, and that remodeling produces changes in the triggers and the substrate. We talked about rotors, and now we know that these rotors are actually, in general, they are initiated in one of the pulmonary veins. About 80% of those triggers are in the pulmonary veins, and there is, in paroxysmal atrial fibrillation patients, there is success in ablation by isolating these pulmonary veins to prevent this propagation of waves that find a heterogeneous substrate and may break the same way as water waves break in the formation of rotors and eddy formation. And these rotors, once initiated, they can result in very complex activation. They will spin at very high frequency. And again, I must stress that the frequency is the important parameter here. They generate electrical turbulence. The higher the frequency of this rotor that is located here, the greater the turbulence will be in the overall atria. In fact, if I were to reduce the frequency of this rotor by, for example, reducing IK1, just to say something, I will convert this fibrillatory pattern into a flutter-like pattern. And so frequency is the problem here. Flutter and fibrillation have the same basis. They are both rotating waves. In the case of flutter, the rotor is anchored in the right atrium, for example. The typical flutter is anchored around the crista terminalis and propagates from the sinus to the AV node. But now when these rotors are maintained, imagine these rotors maintaining for long periods of time. And they can maintain for years and for the lifetime of an individual. So what happens to the atrium when this fibrillatory activity happens? What is atrial remodeling? Well, atrial remodeling is a very complex problem. It is multifactorial and it is extremely complex. Well, let's think that we start with high frequency excitation of the atrium. Let's do an experiment in which we take a sheep heart and apply stimuli very rapidly, and we excite it for a period of an entire year and produce fibrillation for the period of an entire year. This sustained high frequency excitation is going to produce changes in the myocytes, is going to produce changes in the fibroblasts and in other cell types. And it is going to produce stress, and that stress is going to produce oxidative stress with the production of ROS. And these reactive oxygen species can have very significant effects on the ion channels. In addition, the atria dilate, and they dilate because they cannot contract. And eventually they dilate more and more and more. And then, in addition, there's an inflammatory response directly to the high frequency excitation. The myocytes themselves generate cytokines, generate proteins that attract the inflammatory cells to the heart. And all of these three factors work in concert and they work at different times. And we can see these as pathways, as very complex pathways that are given by these colors. You have the oxidative stress pathway, you have the atrial dilatation pathways, you have the inflammatory pathways, and all of this produces changes in gene expression. And these changes in gene expression are going to produce changes in the myocyte, et cetera, et cetera. And they're going to result in fast changes and slow changes, and intermediate rate changes. Let's just very rapidly look at the fast changes. Among the fast changes, I mentioned oxidative stress with the production of ROS. At the membrane, there's oxidation, and that oxidation produces reactive oxygen species which are going to attack directly the ion channels and are going to produce something else. They're going to lead to calcium overload. So the fastest changes that you will see are the reduction in the calcium current and the activation of the L-type calcium channel. And those are going to shorten the action potential duration. Now, there's something interesting that Dobry told us, that in his patients, the myocytes did not seem to have short action potential durations when isolated. But this does not depend on gene expression. This depends on a very rapid effect of the reactive oxygen species of the ion channels, which is reversible. And we do know that if we stimulate very rapidly the atrium, the action potential is going to get shorter. But if you stop that stimulation, the action potential is going to return to control. So this does not depend on gene expression changes. These are post-translational modifications. There is also calcium overload, which Dobry very beautifully told us about the formation of VADs and trigger activity. And in addition, there is apoptosis, myocyte, cell death. And all of these changes are fast. These are intermediate changes, and together they lead to rotor formation. And that rotor formation eventually, because of the high frequency excitation, is going to lead to the formation of fibrosis, And these two, calcium overload plus fibrosis, plus activation of cytokines, are going to lead to long-term electrical structural remodeling. Now this scheme is oversimplified. There are many, many changes that are occurring within each one of those colored pathways. Imagine these are networks of thousands of proteins that are interacting with each other, and they are interacting within pathways and between pathways. So, sustained high-frequency excitation leads to oxidative stress, which shortens the action potential duration, and also produces calcium overload, which leads to trigger activity and rotor formation. And that also is associated with atrial dilatation, with neutrophil recruitment, angiotensin II recruitment, inflammation, myofibroblast activation, TGF-beta, fibrosis, and long-term electrical and structural remodeling. That's basically, in just one slide, what remodeling can be, but it involves thousands and thousands of proteins. So in order to start understanding this, we use a tachypacing-induced model of persistent atrial fibrillation in the sheep. These are not humans, but it is a very interesting model because its remodeling resembles clinical remodeling in persistent atrial fibrillation, including atrial dilatation, fibrosis, and electrical remodeling. So we insert a pacemaker into the right atrium, and this pacemaker is modified to apply bursts of rapid pacing during 30 seconds at 20 hertz, and then stop, and if atrial fibrillation is present, the pacemaker will not resume. As soon as sinus rhythm returns, the pacemaker will reactivate itself, and so on, to do what atrial fibrillation begets atrial fibrillation does, and that's what Ricardo was telling you about. So we do see all of these changes, and what we see very nicely is that if we plot the episode duration, the percentage of episodes here in the y-axis, and the weeks after pacing on the z-axis, you will see that in this particular animal, the first episode started at 4 weeks, the first spontaneous episode after burst pacing. And there are tiny little episodes, so there are many, many, many tiny little episodes here. And the number of episodes diminishes, but their duration increases progressively more and more and more until at week 14, this is persistent atrial fibrillation. And it maintains there for a year or longer, the duration of the experiment. So this is the protocol. We pace, start pacing, and then the first episode occurs approximately at 5.5 days. Then if we continue pacing, we see what we call paroxysmal atrial fibrillation, and this lasts about 7 weeks, and then all of a sudden we get persistent atrial fibrillation, and at this time no more pacing, fibrillation is self-sustained, and this usually takes about 55 days. So it's a long-term experiment, and we keep the animals for more than 12 months, and then we study 3 different groups, what we call sham-operated animals, which are controls, animals in the transition, and the long-standing persistent atrial fibrillation animals. That is, the comparisons we make are always having a control. So what are the immediate consequences of sustained high-frequency excitation in the atria? I told you that the immediate consequences is the liberation of ROS. We haven't studied the ROS in our laboratory, but several laboratories have studied, particularly Barbara Casadei in London has been studying the role of ROS in remodeling, and it has a very important role. But what we see is that we do see the consequences of that ROS activation. The reactive oxygen species lead to electrical remodeling of the short term with reduction of the L-type calcium current, increase of the IK-ATP channel, reduction of the action potential duration, and this, together with trigger activity, permits rotor formation. So, we do know that the refractory period shortens progressively at the start of fibrillation or at the start of pacing. And this, it shortens very rapidly within 1 or 2 days after the onset of pacing. So these changes are probably reversible changes that occur in the absence of gene expression changes. They occur directly at the ion channel level. And they can last for as long as you maintain the high frequency pacing. But as soon as you stop them, if you stop within the first 2 days, this is going to reverse. But there are times at which it will not reverse. So how does electrical remodeling affect rotor frequency and stability in the sheep model? Action potential duration shortens, and it also, in our model, it shortens both during the transition and when AF is persistent. So there are two factors occurring here. One factor is the rapid effect that Ross has on the ion channels. And the other factor, as we will see in a moment, is gene expression changes. In both cases, what is maintained is the shortening of the action potential. Here we have superimposed shortening of the action potential during the transition and in the persistent. And in both cases, the action potential are extremely short, and the action potential does not depend very much on the frequency at that time. It is frequency independent because it has to be shortened to the maximum. Now, how does it happen? Well, we have made a very interesting observation that I think may have some clinical importance. So, if you look at the first episode that occurred within the first 5 days or so, you will see, and if you measure the interbeat interval and then do a fast Fourier transform, you will see that it's 7.3 hertz, 7.3 cycles per second. But if you wait until AF becomes persistent, that frequency goes up to 10.3 hertz. And if you wait 4 months, the frequency has just a little bit higher. So if you plot now the time course of the dominant frequency change as a function of the time after pacing, you will see some very interesting results. This animal, for example, started with its first episode at 1 week, and it was 7.5 hertz. The next week, it had increased to 8.5 hertz or 9 hertz, 9.5, and then 10. And then all of a sudden, at the arrow, it no longer changed. At this time, it was persistent atrial fibrillation. This animal was slower. It took 4 weeks for the first episode to come out. And then, as the episodes became longer and longer, the frequency increased progressively up until the 9th week, at which time AF became persistent. So, if you now plot the slope of this line, this one, as a function of time to start persistent pacing, all of these animals fall on the same curve. Actually, you can do a log-log plot and you get a straight line and they all fall in the same curve. The difference is that some animals take just a couple of weeks and other animals take up to 30 weeks. We don't know why, but there is this thing that gives us the idea that we might be able to stratify patients to see when they are going to become persistent. If you take a patient that just came to your office and started with his first episode, what if you treat them with something that prevents fibrosis, for example, and treat them chronically to prevent that fibrosis? The atrial is not fibrotic as yet, probably, or if it is fibrotic, it's not as fibrotic as if the AF were persistent. Perhaps you can make some changes there. Importantly, always the frequency in these animals remains, the highest frequency is always in the left atrium, always in the posterior wall of the left atrium, the majority of cases. And that's another issue that correlates to the clinic. Most of your patients, you do ablations in the posterior wall of the left atrium, around the pulmonary veins or near the pulmonary veins. These rotors have the tendency to go to the pulmonary vein region because that's where the gradients of the action potentials are located. That Ricardo Caballero was talking to us about, the gradients in IK1, the gradients in other ion channels are there. So, ask another question. Does the transition from paroxysmal to persistent atrial fibrillation result from dysfunction of intracellular calcium regulation? There is no question that there is calcium overload here. The more frequently you stimulate, the greater the overload of calcium. But what happens to the proteins that control calcium? And those proteins are similar to the same proteins that Dobry was telling you about. Now I'm talking about the remodeling process. Are these important in the long term? And we were not able to find significant changes in many of these proteins. They either decreased or they were unchanged. We saw that SERCA was unchanged, was decreased, phospholamban was decreased. The only one that increased was the sodium-calcium exchanger, probably because of the calcium overload. And the CAM kinase did not change in our hands. Moreover, the ryanodine receptor in our hands was decreased and its phosphorylation was unchanged. So we didn't see any of these changes in this chronic animal of atrial fibrillation. My guess is that in this animal, there's no heart failure. And in many of the patients where we take myocytes, they do have heart failure in those patients. So there's a different. We are talking here about lone atrial fibrillation in the absence of other comorbidities. Now, so we concluded from this that while intracellular calcium overload occurs in this model, the transition from paroxysmal to persistent atrial fibrillation does not seem to depend on calcium leak, DADs or EADs, and this does not lead to trigger activity. In our model, there does not seem to be trigger activity in persistent atrial fibrillation. And it makes sense for us because there is calcium overload, but the transients for calcium are very tiny or very small. So, what's the next step in this? There seems to be refractoriness in the ryanodine receptor, and there seems to be downregulation in its phosphorylation, and therefore the DADs and EADs are not playing a role in this model. That doesn't mean that they don't play a role in other models. That doesn't mean that they don't play a role in patients. But in this model of lone atrial fibrillation, the ryanodine receptor does not seem to play a significant role. So, let's move on a little bit. Which ion channels explain the transition from paroxysmal to persistent atrial fibrillation? Ricardo already talked about some of the ion channels, and Sandeep also talked about some of the ion channels and the role in rotor formation and maintenance. Well, in this model, we see a significant reduction, long-term reduction of the gene that codes for the L-type calcium channel, the gene that codes for the sodium channel as well. And this happened both in the right atrium and in the left atrium. So the reduction in the calcium current not only is important early, but is very important late as well. There's also, at the level of the protein, when we do Western blot analysis, the calcium, L-type calcium protein is downregulated, the sodium channel protein, the alpha subunit of sodium protein is also downregulated both in the right and the left atrium. So here we have two very significant effects that makes us think that the action potential duration is shortening and the excitability is going down. And these we complement with the recordings of the ion channels themselves, and indeed the gene expression, the protein expression at the membrane, and the ion channel itself, the current itself, are both all downregulated for both L-type calcium current and the sodium current. There's another change that has been commented about, and that's the ITO. The ITO is downregulated. So ITO downregulation would work against the shortening of the action potential. We want to prolong the action potential duration. So ITO does not seem to play a role in the shortening of the action potential. And on the other hand, IK1 is increased, as was increased in the patients with atrial fibrillation, and that contributes to the shortening of the action potential. So we have three major factors here: this decreasing of the sodium current, decreasing the sodium current, decreasing the L-type calcium current, and increase in the IK1. Notice, these are changes in gene expression. Some genes go up and other genes go down. This is not that everything is blunted. Here the heart is reacting to the stress produced by the high frequency excitation. So, we come back to our diagram, and now what happens next? Well, there are long-term changes in transcription of ion channels, and this is what I just showed you before. So can these changes really explain what we see when the frequency increases? So why don't we do a computer model the same way as Sandeep showed you. Actually, Sandeep did this experiment with some collaborators. So change the ion currents and just reduce the calcium current, increase ITO, and let's see what happens. And indeed, when you do that at the level of the action potential, what you see is if you only reduce the L-type calcium current by 30%, as we saw it in the experiments, you will see a shortening of the action potential. But if you now combine reduction of the IK1, increasing IK1, reduction in L-type calcium current, and reduction in sodium, what you see is a significant acceleration of the rotors because the action potential duration has hyperpolarized slightly and the action potential duration has shortened. So these are the three major ion channels that correspond to the remodeling of rotors in atrial fibrillation. We see a progressive increase in the current, in the dominant frequency, and that is because initial changes in L-type calcium current and then progressive increases at the gene expression level of L-type current, sodium current, and IK1 changes. Let's talk about atrial dilatation. That's another, we talk about electrical remodeling. So what's happening at the level of the structure? This model develops significant atrial dilatation. And these are two posterior walls of two animals. And David Filgueira did this beautiful picture. These are 63 kilogram sheep, one 63 kilogram sheep and one 65 kilogram sheep. And you can see that the remodeled atrium is very, very significantly dilated. But these animals, this is control, this is persistent AF, they do not develop heart failure. The left ventricular ejection fraction is normal, and there is some mitral valve regurgitation, perhaps because of the atrial dilatation, but significantly the left atrium is highly dilated as is the right atrium. So what is the effect of atrial dilatation, of the stretch on rotor frequency and dynamics? Well, many years ago, Jerome Khalifa, also in the laboratory, did some beautiful experiments in which he did took the sheep heart, isolated, Langley nor perfused, and increased the intra-atrial pressure hydrostatically by just filling it with thyroid solution. And this is an experiment that was initially done in the laboratory of Mauri Salesi in the rabbit, so Jerome just transported it to the sheep. And what we could see in this experiment was that as soon as you increase the intra-atrial pressure, and he was measuring the dominant frequency, and this is the plot of dominant frequency, 6 hertz in this and 15 hertz in red. What he saw is an area very close to the pulmonary veins in which the frequency went way up to 15 hertz. And he also demonstrated that the waves were propagating away from that region, suggesting that the rotors were located, the dominant rotor was located there. And measuring the dominant frequency very nicely, did the quantitative analysis by increasing the intra-atrial pressure to more than 10 centimeters of water, the dominant frequency increased dramatically. So stretch shortens action potential duration, accelerates rotors. And in fact, it also stabilizes the rotors. This is an experiment of Mashi Yamasaki. I think, David, you were involved in that study as well, and I think this is your picture, or is it Mashi? What happens there is that if you have persistent atrial fibrillation, this is paroxysmal, persistent atrial fibrillation. With stretch, the rotors remain always in the same place. If you reduce the stretch, the rotors meander around and eventually terminate. So stretch increases the frequency and increases the stability of the rotors. Okay, let's move on to fibrosis. We see here that the neutrophils that are recruited into the atrium secrete a number of cytokines, particularly angiotensin II, TNF-alpha, and IL-6, and this leads to an inflammatory response which activates the myofibroblast and leads to the release of TGF-beta1 and PDGF. These are two of the most pro-fibrotic molecules there are, and these pro-fibrotic molecules are particularly heavy in the atrium. There is demonstration that if you overexpress TGF-beta in a mouse, you will produce very significant fibrosis in the atrium without changes in fiber in the ventricles, or minor changes of fiber in the ventricles. So TGF-beta actually would contribute to the structural remodeling. And there is indeed evidence that TGF-beta and PDGF, both of them, produce changes in electrical remodeling. So it is the calcium overload together with the myofibroblast activation which is responsible for both the structural and electrical remodeling. This is oversimplified. There are many, many molecules contributing to this effect. But nevertheless, this gives you a very good picture of what's going on. Fibrosis increases progressively in this model. Here, I'm measuring the serum levels of procollagen type 3, which is a marker of cardiac fibrosis in the sheep heart model. And as you can see, when you normalize it to the control, in the sham-operated animal, there are no significant changes, but there's progressive increase in these markers as the animal becomes more and more persistent in atrial fibrillation. And of course, if you take the tissue at the end of the experiment and compare the sham-operated animal with the transition and with the persistent atrial fibrillation animal, you will see a significant increase in fibrosis. So, does the presence of fibrosis modify the complexity of the rotor? How does it do that? Well, we don't know, but we can guess. We took several years ago a model in which we isolated pieces of the atria, made slices, and then digitized those slices for control sheep and for sheep with fibrosis. And then we said, okay, let's put ion channels there and let's see how the rotors behave. So we had two models, one just of the posterior left atrium, and another model in which we included one of the pulmonary veins to see what the role of the pulmonary veins be in the dynamics. And so we, in the control, in the absence of very significant patchy fibrosis or interstitial fibrosis, this is just a tiny amount of fibrosis, we were a control animal, we were pacing at this point and looked at the wave propagation in this model. And, sorry, I have to go. Oh, I'm sorry, I have to go back. I want to show you the control first, otherwise. So, if we pace in this animal, the atria will be very nicely behaved. Remember, this is a very thin slice, but nevertheless, the atria behaves normally. It responds naturally to pacing, normal conduction velocity, and so on. But if now I start pacing an animal with fibrosis, in a model with fibrosis, totally different results occur. So now, if you look here, it's eventually stabilizing around this fibrotic area. The fibrosis seems to stabilize the rotors. Actually, in the next experiment, the the wave anchored around this fibrotic area. And it stayed there for the duration of the simulation. So fibrosis helps to anchor rotors. And we see that in animals who have persistent atrial fibrillation, the rotors seem to be more stable than in animals that are less fibrotic. So it seems that fibrosis itself is a factor that contributes to the stabilization of rotors. So what if you want to prevent fibrosis? So if you want to prevent fibrosis, you will not necessarily get rid of the rotors, but you may reduce their anchoring possibilities and reduce the possibility that the animal would go into persistent atrial fibrillation. What happens here? Well, if you have the pulmonary vein, in the absence of fibrosis, you have non-sustained rotors that eventually touch a boundary and they, boom, terminate. But what happens when you have fibrosis? Well, it becomes more complex, obviously. And now you see that not only the fibrotic areas, but the pulmonary vein itself becomes a center of rotation. And it becomes much more complex, obviously. So, let me conclude by saying that rotors are an important mechanism of AF maintenance, that the sheep model of long-term persistent atrial fibrillation recapitulates the structural and electrophysiological remodeling that occurs in patients, that while intracellular calcium overload occurs in this model, the transition from paroxysmal to persistent AF does not depend on DADs or EADs, and that there are differential changes in sodium, calcium, and potassium channel expression and function that explain the increase in the dominant frequency that occurs during the transition, that atrial dilatation contributes to rotor acceleration, and that fibrosis provides anchoring for sites for rotor leading to AF perpetuation. And these are the guys, some of the guys who did the experiments, and I want to thank all of them, and thank you for your attention. And I'm sorry I went 2 minutes beyond time. Thank you. I'd be happy to answer one question, if you have it, while Dr. Berefeld comes to the podium. One question. Yes?
Well, my name is Fabiana Skornik. I'm from the Cardiovascular Genetics Center here in Girona. My question is, with all this remodeling in an area where you have the highest density of intracardiac ganglia, do you have any thought on, I mean, wouldn't you expect that these would be remodeled too, and that will affect?
Ganglia are connected only at the nerve terminals to the atria. So those nerve terminals would go to their point of contact with the myocyte, and yet they would be remodeled. And they would affect even the ventricles if there is remodeling of the nerve. But I don't believe that would be a local response to the ganglia. Because those ganglia are connected to the nerve terminals. The nerve terminals are the ones that are the effectors, not the ganglia themselves. The ganglia are just sitting there. They are the connections to the atria are the ones that are important there. So I believe that if these nerves are heterogeneously distributed, those nerves will modify differentially the atria. It's possible that nobody really has studied it in detail. Pausa has done some of it, but it's possible that the left atrium receives most of those nerve terminals. But again, it is not the ganglia that are playing a role here themselves. The ganglia are the brains of those nerve terminals. You have to think in terms of the effectors themselves, and those are heterogeneously distributed. Right? The cell bodies are there, but the cell bodies have axons, and those are the ones that are the effectors, right? Okay, let's move on to Dr. Bernfeld, who's going to talk to us about high-resolution mapping in AF. Now we're going to get closer to the clinical situation, and he's going to talk to us about some of the pitfalls that may occur when one tries to do high-resolution mapping. Omar?
Thank you very much.
This is the pointer. Yeah, and you can actually go forward, backward. This is the pointer.
Thank you, Dr. Khalifa. I would like to first thank the organizers for inviting me to speak here. It's indeed an honor, a privilege, and I'm delightful to be here and speak to you. Much of the work that I'm going to talk about was actually done in Spain, and I have very fruitful collaborations with people in this country. I really enjoyed this visit. So my talk is a little bit changing emphasis. I'm talking about atrial fibrillation, but it's going to be more on the methodological aspects of studying atrial fibrillation, although I strongly believe that as the mapping of AF has two roles. First, understand mechanisms of AF, and then guide ablation procedures to terminate AF, so we can set our objective to study mapping methods and the mechanisms of AF. And as I indicated by these two arrows, these two questions are, of course, not independent of each other, and we have to be very careful in our methodology approaches, because I start with the premise that intuition can sometimes go wrong. And although this picture is absolutely not related to what we do here, sometimes errors in our field can be not so funny as maybe in this picture. Anyway, we should keep ourselves critical of the work. So the outline of my talk is the following. I will cover 4 topics. I will start with background, how we set up our concepts and methods by studying atrial fibrillation in the ship. I will move to patients with simple sequential single electrode mapping that is widely used in the clinic, and then move to 2 developments, recent developments. One is the panoramic mapping using the basket catheter, and the other is going from invasive to non-invasive mapping with body surface mapping. We'll start with the ship. This is our main experimental tool. We isolate the hearts. In this diagram, I'm showing how we set our cameras. We can put up to 3 cameras to map simultaneously from various regions in the heart. One of them is looking with fiber optics inside the heart, and 2 can be positioned on the left and the right atrium. And here I just show some anatomical pictures as seen from the endoscope that looks inside. The advantage of that endoscopic mapping is that the atria remains intact, and we can study the atrial fibrillation in a more physiologically realistic condition. This is a movie, unprocessed movie of atrial fibrillation in the presence of isoproterenol. We can see a rotor, not very clear, but if you look a little bit closer, after some time you realize that there is a rotational activity in the left atrium and more complicated activity in the right atrium. We, for many years, looked at those movies and created activation maps. But then we came up with 2 methods to enhance the understanding of those movies, and the methods consist of basically 2 methods. One was looking at the fixed, what we call stationary properties, and that was done with Fourier transform that looked at the dominant frequency. And the other method was intended to look at the dynamic, or the details of the dynamic propagation patterns, and that was done with a Hilbert transform. And I will explain a little bit about these 2 methods. First, about the dynamic properties. What we do, we perform a transform called the Hilbert transform, in which allows us, instead of presenting the change in time of transmembrane voltage, so instead of coloring the voltage itself, we now color code the action potentials based on its phase as repolarization, upstroke, plateau, and then resting. These colors are, of course, only cartoon, but this type of transformation is very useful in highlighting center of rotations that we term singularity points or phase singularities, that shows in one frame not only the situation of the front and tail, but also the notion of time progress, because we know that the front, in this case I think it's a blue-purple boundary, it will move only to refractory area, to resting area. So we can immediately superimpose the area, the direction of propagation in these snapshots. Now the other method looks at more stationary properties, not on instantaneous activity, but rather what happens over a period of time. Here I show the snapshots of the movie, and I superimpose that grid to indicate that each pixel is a time series, basically of fluorescence in our case, not voltage. And here I have, in the left atrium, in this pixel, I have fast activity. In this pixel, I have slower activity. And when we apply Fourier transform, we obtain the power spectrum. And the peak of the power indicates what is the frequency that we call dominant frequency. And then we color-coded it into a map to create dominant frequency maps. And here, in this case, this is movies that were taken by David Filgueiras in a camera that was pointing inside. This is the posterior left atrium obtained with an endoscope, and this is the left atrial appendage. And we can see a rotor that moves. It sometimes appears in the posterior left atrium, sometimes, most of the time, in the left atrial appendage. So that gives us an indication of the dynamic process. And here in these panels, I show snapshots indicating that dynamic process. And in the bottom, what I'm showing is a more stationary properties. What happens when the rotor was placed in the left atrial appendage, we found that the dominant frequency was highest in left atrial appendage and a little smaller in the posterior left atrium. The difference is not very large, but our method can detect those changes. And importantly, even that small change is significant in telling us where the rotor or a driver, potential driver, may be located. It's also interesting that when the rotor abandoned the left atrial appendage, that frequency went down to 7.4. So here we have a method to analyze the movies, the optical data in the frequency domain, in the phase domain, to gain enhanced insight. Moving to several questions that came about the third dimension, what happens in the thickness of the wall? One of the things that we could do with optical mapping is map simultaneously from endocardium and epicardium. And here we see two examples, one in the posterior left atrium, pulmonary veins, and the other in the left atrial appendage, when we identified singularity points on both surfaces. And we don't know what happens inside. This line is an artistic rendering of what we believe happens. There may be a scroll wave that extends between the two surfaces. We call that line a filament, and that is the axis that there is nothing there functional, anatomically. There is only functional behavior there. There is only rotation of action potentials around that line, that hypothetical line, and that concept is going to be important later on when I'm going to talk about non-invasive mapping. Nevertheless, we don't always see singularity points on the surfaces. Sometimes we see breakthroughs. This is another example where we see breakthroughs. In this case, those were simultaneously in the epicardium and endocardium, and we don't know what happens inside. And it may be that there is an ectopic activity giving rise to those breakthroughs, but as indicated already several times, it may also be intracardiac, intramural reentry. And I'm showing again the simulations that Dr. Khalifi showed in the previous talk, however, with a different emphasis here. I'm not so much interested in what happens with the fibrosis, but rather the fact that if you look at this scale, it's hard to see, but this is 1 millimeter, suggesting that this thickness is about 3 to 4 millimeters, and we simulated activity there that shows that even in that tiny tissue of, this is a sheep atria, we don't have, at this time, we didn't have the kinetics of the sheep, we implemented the kinetics of a dog, I believe it was a dog. Nevertheless, we were able to see that in very thin tissue we can have re-entry. So there goes the theory of the minimal, the critical mass requirements. Once you have rotors, it can accommodate itself in a very, very small area. Actually, there was data from Mandapati that was published when we started to look at atrial fibrillation. He measured the core sizes, the areas that are covered by the singularity point meandering, and he found in the sheep, those meandering can go as small as 2 millimeters. Indeed, indicating that they can accommodate in very, very small tissue. So to summarize those experimental part of the work experiments, we introduced two concepts, actually three. One of them regarding the rotor is the phase singularity that is obtained in the phase domain. We introduced the concept of filament in 3 dimensions, that's the extension of that singularity point in the thickness of the wall. And we also introduced the concept of dominant frequency, which is the highest power for a period of time, typically 5 seconds. And we summarized that the presence of rotors at the highest dominant frequency sites that drive atrial fibrillation is confirmed in optical mapping in the SHIP model of paroxysmal and persistent AF. I would like to switch now to the patient to show a little bit of data on sequential mapping. This is early work we did with the collaboration of the group of Michel Seguin in Bordeaux. The main investigator there was Prash Sanders. We looked at about 120 points in paroxysmal and persistent atrial fibrillation patient. And the colors here indicate the dominant frequency in each of the points, with interpolation, of course. And one of the findings was that the highest dominant frequency are distributed differently in the paroxysmal versus the chronic atrial fibrillation. In this example, we see the highest dominant frequency near the right inferior pulmonary vein. Typically, in those patients, paroxysmal atrial fibrillation, the highest frequency was in the veins. However, in the chronic atrial fibrillation, those frequencies not only tended to be higher, as indicated by this, the different scale, they tended to distribute differently. There was no preference for the highest dominant frequency to be in the pulmonary vein. They were equally found anywhere. So clinical studies have confirmed the existence of hierarchical organization of rate of activation in different regions in patients with paroxysmal or chronic atrial fibrillation. But of course, the question that is important for us is those fastest regions. Do they have any role in maintenance of atrial fibrillation? And I'm going to just show a few examples to show that indeed they have. This is from the same study by Sanders from Bordeaux. When he ablated the highest dominant frequency site, we saw reduction in the cycle length as measured in the coronary sinus. When we ablated non-high dominant frequency site, there was no change in the cycle length, indicating that the highest dominant frequency had a role in driving the arrhythmia. And this is data from Felipe Atienza from Madrid that mapped sequentially, in this case, the atria are showing the highest dominant frequency near this vein, and these points indicate the isolation of that area that terminated atrial fibrillation, transformed the air heart into sinus rhythm. So we concluded about those patients with sequential mapping that it is the abolishment of pre-existing dominant frequency gradient. It's when you eliminate the highest dominant frequency, you could equate all the areas. Now they will have always the same dominant frequency. It's not necessarily converting them into sinus rhythm, but what was important is to abolishing those DF gradients, that would predictive of AF, freedom of AF in the follow-up. And this is another example where he isolated a highest dominant frequency actually in the right atrium in this case, after isolating the veins that were not the highest frequency to begin with. And I would like to switch now to sequential mapping. This is one of the latest development in the field of mapping and ablating atrial fibrillation. This is data from Sanjeev Narayan's, I think it's the first case report. So he deployed a basket inside the right atria in this case, and he was able to map, in this case, counterclockwise rotor, and upon a brief ablation of less than 30 seconds, that atrial fibrillation transformed into sinus rhythm, showing not only that he claims that there is a rotor there, but it's certainly an area that was important for the AF, because when he ablated it, AF stopped. He then reinduced atrial fibrillation in that patient, and he was able to reinduce after efforts, and then he mapped the left atrium, and he found another rotor, in this case here, again, counterclockwise, and in this case, a little longer ablation, also the ablation was targeting the center of the rotor, and that atrial fibrillation stopped again. He then summarizes many patients, 101 patients actually, in a paper published in JACC, and he showed that sometimes he finds rotors, in this case clockwise rotors, sometimes focal activity. In 70% he actually found rotors, in 30% they were focal. He doesn't know whether they were indeed ectopies, or we could maybe think that maybe intracardiac or intramural, sorry, not intracardiac, intramural reentries. The point is that after brief ablation, or on average 5.5, those AF transformed into sinus rhythm. So we ask ourselves whether those mapping, what is the properties of that basket mapping? Can we learn about the mechanisms of AF with the basket mapping? To have a first suggestion of what happens in those mapping, we run simulations. Javier Saiz and his team ran that in Valencia, and they took the geometry of the electrodes on a basket catheter and incorporated them in a model of the atria containing the left atrium and the right atria. And here you're going to see the simulation of atrial fibrillation in that model. That simulation we know will depend on rotors, because that by construction how that atrial fibrillation was induced. So we have those rotors, and you can see the frames, the splines and the electrodes. And now let's look at the data. This is a summary of his work. So the simulation itself consisted of two rotors that drive the arrhythmia. This is posterior view and anterior view. We can see that there's rotor 1 and rotor 2. The topology rules is that when you have a surface, you cannot have a single rotor in that surface. There must be two rotors. So that is consistent with that theory, and we believe that also in patients, a single rotor cannot live without a pair somewhere. It could be a slower rotor that is changing its location, but there must be always two rotors. So this is what we simulated, and then he calculated the maps on the basket in two situations. The first one is when the basket was making partial contact with the wall. The baskets were retained as spheres, and obviously they were not able to contact the wall everywhere. And the other situation is when the splines of the basket were projected onto the walls to make full contact. And the mapping results were different. What you can see that when you have partial contact, you can identify, this is only a snapshot of the phase movie created by the basket. You can indeed see singularity point here that correspond to this location of rotor 1 and the singularity point corresponding to the second rotor. And that situation is a little bit more fuzzy when you make full contact. Not only we see two singularity points that lasted for a long time and may be related to the singularity, to the rotor 1 and 2. We see other singularity points, and I'm pointing few of them in those red arrows. The point I want to make here is that when you make full contact, you actually have false positives, and you have to be very careful in the interpretation of those basket mapping data. So to summarize this part is that there is evidence based on the basket catheter that rotors in human are present and drive atrial fibrillation, but the computer simulation suggests that the low resolution and poor contact basket mapping is capable of detecting rotors, but false positives could not be excluded at this moment. This is an ongoing research. I cannot give you stronger conclusions more than what I tell you here, and I hope in the near future to gain more insight. Maybe we will be able to discern more clearly between the false and true positives. One comment is that intramural rotors can exist in atrial wall with thickness of as low as 3 to 5 millimeters, so maybe focal activity that was seen by Narayan is actually intramural reentry. Now, let's switch to the last part of my talk, the non-invasive mapping. A recent method that has been used is called the ECGI. It's a panoramic non-invasive mapping. It consists of two different procedures. In one, you place a multi-electrodes vest on the patient and record electrical activity in all the electrodes. And second, you run the patient into CT to obtain the geometry of the torso. And then you combine these two pieces of data into a software that performs the inverse solution and then you get the activation on the epicardium of the heart. And with that method, the Kukulic from the group of Yoram Rudi was able to show various activation patterns during atrial fibrillation. In this example, they show rotors, but the rotors in those studies were not very stable. They tend to meander. This is data from Michel Esseguer's group that shows 2 cases in which they did see rotors on the surface of the atria during atrial fibrillation, but those rotors, in contrast to the data from Sanjeev Narayan, those rotors tend to drift, and they were short-living. They sustained between 1 and 7 rotations. And they were, when they ablated the atria in those studies, they didn't ablate those rotors because those rotors were drifting, but they claimed that when they hit atrial fibrillation, when they hit rotors, somehow the cycle length was prolonged. So the atrial fibrillation became more organized. So we went again and asked ourselves, what can be said about mapping from the surface? And we have our own vest that was constructed by a group of investigators in Valencia, Maria Guillem and Andreu Clement, among others. And we have 67 electrodes spanning across the torso, according to this diagram. We couldn't learn much about atrial fibrillation mechanisms because our patients were very heterogeneous. Not only with post-paroxysmal and persistent atrial fibrillation, some of them had pre-ablations and ablations prior to the study, so we couldn't say much about atrial fibrillation itself. We recorded the signal from 67 electrodes. We had to remove the activity from the ventricles, and 4 seconds activity were analyzed both in the phase and the frequency domain. And for validation, we had the intracardiac mapping using the Cartos system. So I'll start with the spectral analysis. So our objective was to investigate whether sites with high frequency activity in human AF can be identified non-invasively. This is an example of a patient with a left to right dominant frequency gradient. The left was faster. On the left, you see the intracardiac data. On the right, the surface data. What you can see is that the dominant frequency in these 3 locations on the inside of the heart match those frequencies on the surface. And this is the map obtained with the carton. This is our map obtained with the vest. You can see that the highest frequency that was in the left superior pulmonary vein translates into a peak in the frequency in the center of our back. So in the center of your back. That's not the site that traditionally you record with standard ECG. When the gradient was the opposite, the right atrium was faster than the left atrium, same validation was obtained, but now the highest frequency on the surface was actually under your right arm, in the anterior lateral side of your right part, the right part of the torso. And when there was no DF gradient, you see there is more or less uniform dominant frequency distribution. That was also the case on the surface. There was no apparent gradient on the surface. When we looked at the correlation between the dominant frequency measured inside and outside, when the left atrium was fastest, on the right atrium was fastest, we saw that most of the highest dominant frequency when the left was fastest were in the left back part of the torso, and when the right atrium was the fastest in the right lateral part of the torso. And here is a quantification of that correlation. If you look at data from inside versus data that outside, there is good correlation from data on the left and on the right. And very importantly, the opposing sides correlation is very low, suggesting that there is not only sensitivity, but also specificity. You can tell whether the highest dominant frequency is in the left and the right. And here I showed that data. We looked at intracardiac surface match on the DF gradient, not the DF themselves. And we did 2 types of analysis. First, we combined all the 67 electrodes. And second, we looked only at the electrodes that correspond to the site of the 6 precordials. What we see is that if we look at all the 67 electrodes, there is good match between what you look at the surface and what you look inside, what you find inside. However, when you look at only the precordial, that correlation is lost, suggesting that you can indeed detect dominant frequency gradients, but you need to use the whole body surface mapping, not only the precordial. And that's the summary here. We then moved to look at the patterns, not now the frequencies, and the objective was to investigate whether surface high-frequency sites represent rotor activity during AF. We were indeed looking to see if we can look at those rotors. This is the first simple case, just to orient yourself. This is a case of atrial flutter, so based on the intracardiac data, we know that there is anatomical reentry, the typical flutter. On the surface, what you see is not one singularity point, but two. As I said earlier, on the surface, on the closed surface, you must have two reentrant activities, and not only that, each singularity point consists of two filaments. They will go into the body and into the cavity, and somehow they will have to reach the surface. So we saw two singularity points, and they are stable in time. These are snapshots in time, and their location is pretty stable. Now let's move to AF. The situation is much more complicated there. These are two snapshots, 140 milliseconds apart. If we don't process the phase movies, this is unfiltered data, the singularity point shifts or moves very erratically from side to side on the surface. However, when we filter the data around the highest dominant frequency that we find, either from the inside or from the outside, they correlate, that singularity point becomes stable. So that's an intriguing finding. Here is a movie to show you that singularity point on the surface after filtering. It does move, but it's pretty stable. In this case, it's in the center of your back, so probably it comes from the left atrium. It disappears for a little bit and then reappears. Remember that we don't map the floor and the roof of the vest, of the torso. So, of course, we are not really panoramic. We have large areas, but not fully panoramic. Anyway, so that's the singularity points after filtering. They look pretty stable. I have to show you whether they're real or not real. This is again the analysis I would like to show you with greater details. We selected 6 points along the map to show what happens with the filtering. The black traces that are hidden behind the blue are the original traces, and each one has its power spectrum. All of them have this highest dominant frequency, 6.65. So we filter those black traces around very narrow band around that highest dominant frequency, and the activity becomes very organized. These are the blue traces with an arrow indicating the reentrant activity, and indeed showing the stable singularity point. And this is the trajectory of the singularity point. And very importantly, in this panel, I show what happens to quantification of the rotors. Before the filtering, they were present only for a short period of time. The numbers are not here, but it's about 8.3%. And after filtering, it jumps to about 73%. And the rotor duration is not very long at our hand. It goes from about 160, if I remember correctly, to about 450 milliseconds or so. So not many rotations, but they present more very often. Here indicating that it disappears from our field of view and then reappears. Of course, we don't know what happens here. It may have been just outside of our vest. So that dramatic effect of the filtering raises the questions of what are we doing here? Is that a numerical artifact? To answer that question, we again went back to computer simulations. We have a very simple model trying to understand the mapping from the surface. Our model consists of a sphere representing the atria in the center, and it is surrounded by a passive torso, and we can look at the potentials on the epicardium and everywhere inside the torso, and of course on the surface itself. We simulated few cases, and I will guide you through those busy slides. First, we simulated the case when we have a hemisphere left atrial property and hemisphere with fibrosis. Again, I'm not interested in mechanisms of AF or fibrosis. I'm interested in simulating various scenarios. So in the left atrium, we have a stable rotor. As we go in time, that rotor in the left atrium is stable. And in the area with the right atrium, the other sphere, the other hemisphere, we have many, many wave breaks with short-living singularity points. As we go from the epicardium towards the surface, what you can see is that the map becomes more and more smooth. You can see that those singularity points basically disappear. And now we are ending up with two singularity points, and that they are pretty stable in time. Now, one advantage of our model is that we can look at the singularity points as you go from the epicardium to the surface. We can identify those filaments that I started my talk with in the animals. And these lines are the filaments in the torso. There is no action potential rotation there, but the extracellular potential in the volume conductor is changing in time in such a way that you have rotations along those lines. So, what we can see is that we gave different colors to clockwise and counterclockwise singularity points. So what we can see is that the singularity point that starts here, this is the counterclockwise rotor, extends all the way to the torso, and another one here. But look what happens here. A singularity point that starts at clockwise, there is a filament that extends into the torso, and it meets the filament of a counter-rotating spiral singularity points. And those filaments will not reach the surface. This is from a different angle. The point is that if you look at the number of rotors that you can detect, the number of singularity points as a function of the distance into the torso and towards the surface, that number decreases. So we are losing the detection of singularity points as we go from the epicardium to the surface. Okay, I will rush. That I'm about to finish. So the hemisphere, now hemisphere is left atrium and hemisphere is right atrium. We have one stable rotor in the left atrium and another wave break at the interface between the left and right. And these left rotors are indicated here on the stable rotor on the surface. If you look at on the filament, you see that the filament, although this rotor is very stable, the filament diverge a lot as they reach the surface. And when you filter that narrow band filtering around the dominant frequency, the highest dominant frequency, those filaments stabilize themselves, and now they are very, they are easily seen on the surface as stable rotors. I will skip that for the sake of time, but we found that that divergent is the result of the overall activity in the atria. But then we have a very important question. Now we have one rotor and two filaments. So which one is the true filament, which one is the phantom or mirror filament? How can we from the surface know which filament represents the rotor that is driving the AF? So we go back to the power spectrum, and what we found is that the true mirror, the true singularity point, has most of the power under the highest dominant frequency, while the mirror singularity point, the phantom one, will not have all the maximum power at that frequency. And that occurs in the simulation, and we extend that to the patients, and using that approach, we were able to determine where are the rotors that drive the fibrillation. So if the case of the left atrium is fastest, it was in the center of the back, and you can see that histogram, that's where most of the rotors were located. In that case, the left atrium is fastest. When the right atrium is fastest, the drift was mostly in the right anterior part of the torso, and this is the histogram showing that site. That matched very well our mapping of the highest dominant frequency. This is the frequency histogram, the highest dominant frequency histogram. So to summarize this last part, I would say that evidence of the presence of dominant frequency gradients and rotors in human AF is emerging in the clinical panoramic ECGI approach and the body surface mapping modalities. But the computer simulations suggest that pivoting waves seen on the body surface, they do originate at rotors that start at the epicardium, but their spatial stability is affected by the whole atrial activity, and the ECGI may have low sensitivity to highest dominant frequency and rotor activity of the epicardium. And the last slide to conclude regarding mechanisms of AF, that the phase and dominant frequency analysis enhance evidence of rotors as often drivers of AF. And regarding guidance of ablation or how we use mapping, the critical point is to have panoramic mapping of highest dominant frequency and phase singularities, but there could be caveats. Regarding the basket mapping, we may have false positive targets, and the non-invasive mapping approach has a caveat of low sensitivity. And with that conclusion, and thanking all the people, the many people that participate in that project, I would like to thank you for your attention.
Anybody has a really, really burning question? Really, really burning question. So you'll have 30 minutes to swallow this thing, and then we'll come back. So the next presentation is, now we're going to start talking about therapy, finally. And whether the therapy that Juan is going to talk to us about is effective, we will have to see. But Juan Tamargo is one of the leading pharmacologists, certainly in Spain and around the world. And I'm really happy to introduce Juan, who's going to tell us about the pharmacology of atrial fibrillation.
Thank you, Pepe, and thank you, Nicasio, for giving me the opportunity to be here. Let me start with this slide. This is a paper that has been published very recently in Spain. I just want to show you this data, but particularly this one, said we have 1 million patients with atrial fibrillation. And do you think that you can perform 10,000 ablations per year? I know that is not true, but I accept today, this evening, that you practice 10,000 ablations. So it means that less than 1% of the Spanish population with atrial fibrillation can be submitted to non-pharmacological treatment. And the question is what we are going to do with the 99.3% of the other patients. And the answer is quite simple. We are going to use drugs. But look at this slide. As a pharmacologist, I can tell you that the only area where we stay 25 years without a single new drug is antiarrhythmic therapy. During this period of time, 8 statins, 13 ACE inhibitors, 9 angiotensin receptor blockers. Only one single compound in Spain, dronedarone, please remember in the US, was not accepted because this compound was able to maintain sinus rhythm, but because reduced hospitalizations, and one compound in Europe, not in the US, and it's vernacular. So I'm like a scratch. I'm just looking and say, well, I can probably in the next 30 minutes I can tell you that there is something new in the horizon. And nevertheless, if you think that this is my life doing research in pharmacology, I'm quite skeptical. I don't want to say cynic, but skeptical with many of the therapeutic approaches that I'm going to present to you. Well, the background is that antiarrhythmic drugs represent the mainstay of therapy, but they present a very modest efficacy, serious adverse effects, and most of them are contraindicated in patients that really need antiarrhythmic agents, those with a structural heart disease. So the objective is, as a pharmacologist, to develop new compounds that are safer, and this is the point, safer and more effective antiarrhythmic agents. And from a pharmacological point of view, if you want to develop new compounds, the first question is, I need a clear understanding of the pathophysiological mechanism directly involved in the genesis and maintenance of atrial fibrillation. This means a well-supported hypothesis, but Professor Khalifa already mentioned we don't know much about this. So we are developing compounds without knowing which are the real targets involved in the maintenance of atrial fibrillation. So the bottleneck is that atrial fibrillation is a complex condition associated with multiple etiologies, mechanisms, and comorbidities that is associated with electrical, structural, neural, and contractile remodeling, and this remodeling perpetuates the arrhythmia, and different comorbidities, and I'm thinking now in comorbidities, means different arrhythmogenic substrates, and of course, different treatments. So this is my agenda. I'm going to present you some compounds that can be interesting from the point of view of the electrical remodeling. So the so-called atrial specific ion channel targets. This means targets ion channels that are exclusively or predominantly expressed in the atria. Atrial selective targets that are presented both in the atria and in the ventricle, but their inhibition produces a greater effect in the atria than in the ventricle. Some comments on the old and new targets. So another concept is to modulate the surface density of functional channels and gap junction modulators. And then I will move to the structural remodeling, the so-called upstream therapy, and finally some comments on the abnormal calcium handling. Well, this slide is just to show you that there are some currents that are particularly predominant in the atria, not in the ventricle. IKS is because of the comment that Dr. Caballero already asked. Let me start with this current. We spend a lot of time with this current. This is the ultra-rapid component of the delay rectifier. We were very interested in this current because it's an atrioselective current. You can see it's present in the atria. It's not present in the ventricle. The first compounds that were developed, the first IQ blocker, they demonstrate that they increase the atrial effective refractory period more in the left than in the right atria, but and also these compounds increase atrial contractility. The problem is, if you see this compound, is that in sinus rhythm, these compounds markedly shift upward the plateau phase, but you can see this activates the potassium alpha currents as a consequence. These compounds in a non-remodeled atria, they are not going to prolong, but to shorten the action potential duration. And in patients with atrial fibrillation, they slightly prolong the action potential duration. So from these 4 panels, it seems that could be a reasonable target. The problem is that IQ density is reduced at fast rates in patients with chronic atrial fibrillation. Second is that IQ inhibition might promote early after depolarizations and atrial fibrillation in non-remodeled atria. And finally, that we have evidence that loss of function KCNA5 mutation produces familiar atrial fibrillation. So at this point, I'm not very in favor of IQ selective blockers for the treatment of atrial fibrillation. Of course, you can look at some compounds like this one. You can see that this is this compound from a British company. You can see that produces a dose-dependent prolongation of the action potential duration in the atria, no effect in the ventricle, produces an increase in atrial effective refractory period, no changes in the ventricular refractoriness, and as you can see, the duration of atrial fibrillation decreases progressively as the drug progressively increases atrial refractoriness. Of course, I have here my friend Dobrev, and this is a seminal paper, as has been already a comment of Dr. Caballero, is just to mention that this agonist and receptor-independent potassium-activated current is atrial and pathology-specific target, because this agonist-independent constitutive current is present in patients with chronic atrial fibrillation or after atrial tachypacing. The problem is that we must develop compounds that selectively inhibit this current independently of the normal conventional IKSCH to avoid cardiovascular, gastrointestinal, and central nervous system adverse events. I know that Professor Dobrev, these 2 lines come from his lab. If there's any question, I will just push to you. Well, there are some compounds, as you can see here. Once again, this is NTC801. No, this name disappeared because it has been swallowed by an American company, so this Japanese name disappeared. You can see with these compounds there is no effect on the atria, there is no effect on the ventricle under normal conditions. Now if we add In this case is carbachol. You can see that carbachol activates the acetylcholine, activates the potassium channel. There is a marked shortening of the action potential duration, and you can see that with this compound we can reverse this effect. You see the marked shortening of the action potential duration induced by carbachol is progressively counteracted when we increase the concentrations of this selective compound. Sorry for this slide, but it's very important for me. This is what we have been developing in the recent 10 years. A lot of compounds. We said this is in yellow, means compounds that are specific and selective blockers of IKCH. But you can see that most of the compounds, several comments. First, there are many holes. I don't know what happened on these currents with these compounds. Second, most of the compounds are not selective. They block a lot of currents at the same time and the same range of concentrations. So, and also many of these compounds were studied in single isolated cells or in human clone channels. So it's a real problem, you know. And the problem for me is the following, okay? Let me see how many clinical trials. And the point is, look at the drugs. Do I know something about the pharmacodynamics? This means these compounds are selective channel blockers, or they can block the channels not only in the heart, but in the brain, for instance. I'm thinking in vessels, central nervous system, and immune cells. I don't know nothing. What about the kinetics? I don't know nothing. What about safety? I don't know nothing. Because all the data came from experiments in single cell or in animal species where we are not looking for adverse effects. And now let's see some of the development. Phase 2, discontinue, I don't know. And then look, phase 2 that finished in 2012. Where are the data? Where they were published? Nowhere. So what is happening with all these compounds? And the question is, we have a lot of work in these 2 slides, but I don't know how much of this information I can put on the table ready to be used in the next 10 years. Of course, there is another area of interest, is the so-called atrial selective sodium channel blockers. Atrial cells present several characteristics that differentiate this atrial cell from this ventricular cell. The first is that the resting membrane potential is more negative in the ventricle than in the atria. Second, as you can see, is that you can see there's a slower phase 3 in the atria than in the ventricle. And this is an important point, and is that this is the ventricular inactivation curve of the sodium current. This is the atria, and you can see this, that the half inactivation voltage of the sodium current is shifted in the negative direction by approximately 12 millivolts. What does it mean? This means that at the resting membrane potential, on the normal conditions, at this resting membrane potential, there are less rested but more inactivated sodium channels in the atria than in the ventricle. And so it's possible, based on this, that we can produce a selective blockade of the sodium current in the atria and not in the ventricle. And probably the best example is this one. This is ranolazine. This is a compound. This is the only compound probably of all what I'm going to present to you that probably in 3 years will be ready for approval or approved if lucky enough. And you can see here that ranolazine prolongs the action potential duration in the atria but not in the ventricle, shifts the inactivation curve to more negative values, so there is less availability of sodium currents in the atria, but the normal availability of sodium channels in the ventricle, and at fast rapid activation as in atrial fibrillation, you can see that the atria depolarize, but we cannot reach the maximum diastolic potential because the next action potential appears, and as a consequence there is a progressive progressive accumulation of sodium channels in the block, in the inactivated state. At the same time, this does not happen in the ventricle. So from this point of view, we know that with ranolazine, we can produce a much greater rate-dependent sodium channel block in the atria than in the ventricles. And indeed, this marked blockade also is accompanied by a post-repolarization refractoriness. Let me skip it. There's another interesting point with this compound, is that ranolazine can, these are normal activity and trigger activity induced in the superior vena cava sleep preparations, and you can see that the normal automaticity is suppressed by ranolazine. Early and delay after depolarization are also suppressed. And this is very important because we always talk about fibrosis, inflammation. Please do not forget ischemia. And during ischemia, this is a very simple experiment, and you can see that in ischemia there is an alteration, a failure in the inactivation of the sodium current, and we have here the late sodium current and increasing intracellular sodium concentration, the activation of the sodium-calcium exchange, and this results in an increase in sodium and calcium, a sodium and calcium overload that can produce cardiac arrhythmias. This process is also inhibited by ranolazine. Professor Anselevich did these experiments, and he said that if he combined ranolazine plus dronedarone, or instead of dronedarone, amiodarone, he can produce a much marked prolongation of atrial refractoriness with this combination than with ranolazine or dronedarone alone. Well, this is a nice idea, and this is what is tested in the Harmony. Harmony has been already closed. The data are ready to be published or presented in the next future. And this is the only compound, ranolazine, where I can present to you that there are studies running and recruiting patients. I just want to talk a little on IK1. This slide has been already presented by Ricardo. IK1 is critical for the maintenance of the resting membrane potential action, potential duration, and refractoriness. And we hear this afternoon several times that IK1 increases 2-3-fold in atrial fibrillation, and so it increases rotor frequency and facilitates the stability of the rotors. Some people, including our lab, we believe that ventricular IK1 is mainly carried by KIR 2.1. The atria is more important, KIR 2.2 and 2.3. And just want to present this data, not to propose you that propafenone is a selective blocker or so. No, it's just to tell you that it's possible to start with the pharmacology of IK1. We were able to demonstrate that 2.3 and 2.2 that are mainly in the atria are inhibited at lower concentration than 2.1 that is present in the ventricle. And I just want to present you the next slide, not to be in conflict with Pepe's, to say that we have data that he present with chloroquine. He present data from David Filgueiras, very nice, where you see that chloroquine blocks the pore. What I'm going to present to you is that with flecainide and with propafenone, what we found is that we can modulate the channel and to modulate IK1, but the drug is not in the pore. See, it's not in the pore. It's at the cytoplasmic domain. It's at the cytoplasmic domain of KIR 2.1, and in particular, the propafenone binds to these two residues. As you can see, if we mutate these two residues, the effects of propafenone disappear. So this means that at least we have in the last 4 years 2 possibilities to modulate from the pharmacological point of view IK1. This slide already was presented also by Dr. Caballero, but I just want to add this. He presented that there's an increase in IKS, that IKS is modulated by sympathetic stimulation, that what I want to present to you here is that there is not only electrical, there is no structural, but it's also neural remodeling. And you see here that there is an increase in beta receptors. And of course, this increase in beta receptor is accompanied by an sprouting of sympathetic nerves. And if I like IKS, is because this is a current that don't play any role on physiological heart rates. But when we increase the rate, this current significantly increases. This is the opposite of IKR. IKR presents reverse use dependent. It's very important at slow heart rates. I'm not trying to treat slow heart rates. I'm trying to treat fast heart rates. I'm going to skip this one. So, if you want targets, I can offer you a lot of targets. Forget this. I mentioned this in this question mark is forget about here, propafin. So these compounds with underline are those who have been tested in clinical practice. And as you can see, we have a lot of targets, many targets. I just want to mention this one because we spend a lot of time talking about gap junctions. All these compounds, except this one, have been abandoned. And this one, I just want to present to you to re-emphasize that atrial fibrillation is not a single arrhythmia, a single disease. This roticaptide is effective in atrial fibrillation induced by acute ischemia and induced by, as you can see, mitral regurgitation, but is ineffective against atrial fibrillation associated with heart failure or associated, as you can see here, with atrial tachypacing. This means that these gap junction modulators will be effective in atrial fibrillation associated with a decrease in gap junction conductance, but they will not be effective if we have a reduction in gap junction density or lateralization. So once again, tell me which is the substrate, and you can understand why a drug is effective or not. And this is the point. These are 5 different studies. So, well, my first criticism, these are very young patients. But nevertheless, you see that 60% has hypertension, 20 to 30% coronary artery disease, 30 to 35% heart failure. And you see that aging, hypertension, coronary artery disease, and heart failure are associated with a structural remodeling. Fibrosis, dilatation, hypertrophy, stretch, oxidative stress, inflammation, and ischemia. So the question is, do we have any drugs to prevent this remodeling, structural remodeling, the so-called upstream therapies? Yes. But before, let me show you a couple of slides. This is 3 different models of atrial fibrillation, and you can see that there is very different the amount of fibrosis. What is not shown here is the type of fibrosis, because we can have 30% of fibrosis on the atria, but there are very different. The compact fibrosis is impossible to be reversed by any drug. If you have a sparse fibrosis, things can be different. And this is the slide that changed my mind years ago. It's the paper from Frustacci. These are patients with low atrial fibrillation, and these patients that on theoretical grounds they do not have a structural heart disease. No structural heart disease, they have fibrosis, hypertrophy, and inflammation. So this is a slide where you can summarize cardiac injury, mechanical stretch, oxidative stress, as already Professor Leifer presented to you, growth factors, neurohumoral activation, cytokines, facilitates the transformation of cardiac fibroblasts. They proliferate, migrate, and differentiate into myofibroblasts that again produce endogenous mediators, angiotensin II, endothelin 1, growth factors, and cytokines. And these myofibroblasts, as you can see, produce fibrosis and modulate conduction velocity, refractoriness, ectopic activity, leading to atrial fibrillation. And even when it's not quoted here, there's a very nice paper published by Wu, very recently in cardiovascular research, where they demonstrate that cytokines growth factors, endogenous mediators also, as you know, produce changes on the electrical remodeling. So they produce not only a structural, but also electrical remodeling. And there are some targets here, as you can see, TRPM7 and TRPC3. These are ion channels that facilitates calcium entry and that facilitates the proliferation, migration, and differentiation into myofibroblasts, and can be interesting therapeutic targets. And this is one slide that somebody asked me a couple of years ago. We have a lot of compounds. Once again, underlying compounds are already in the market. And the point is that if we want to develop new compounds, the problem is that we need to develop compounds better than those who already have been studied. ACE inhibitor, angiotensin receptor blockers, or compounds like these two, pirfenidone and tranilase, that are compounds that are used for pulmonary fibrosis, and tranilase for keloid and hypertrophic scar. I'm going to present you just one data published very recently by Nakatari with tranilase. You can see that tranilase is able to prolong the action, the atrial refractoriness, and this is associated with a decrease in atrial fibrillation inducibility and in the duration of atrial fibrillation. What is more important is that this compound is very effective to reduce, as you can see here, interstitial fibrosis tissue. It reduces TGA beta, and as you can see also it can modulate the calcium channels. What about RAS inhibitors, statins, and PUFAs? I want to present you this data, because if you pull the data with ACE inhibitors and angiotensin receptor blockers, you got a significant result as a pool. But I don't want a pool. I want to see what happened in patients. You see, this is statistical significant. But if I look according to the comorbidities, I can conclude that these compounds, ACE inhibitors and angiotensin receptor blockers, in primary prevention are only effective in patients with left ventricular dysfunction or congestive heart failure, but not in patients with hypertension or with myocardial infarction. So this is what I already present to you. And in secondary prevention, these two type of compounds, they are very modest results, only with ACE inhibitors when associated with amiodarone. With statins, the only data are in postoperative atrial fibrillation, and with omega-3, the data are quite contradictory. Why we fail? Know that because we are too late. We are trying to treat atrial remodeling when this process extended beyond the point of reversibility. And second possibility is the limitation and difference on the presently available drugs. So the future is to identify the ideal target population. And second, to understand the different types of human atrial structural remodeling. Do I have any evidence that if we treat early enough, probably we can prevent atrial fibrillation? Yeah. Well, this is a very old slide. This is the normal atria. This is an atrial patient with 1 year in atrial fibrillation. Do you think that it's possible to recover this? And the answer is no. This has been, I cannot recognize the atrial structure. No? But see, there are two papers at least where they demonstrate these are patients that were followed, as you can see, for 3,000 days. These were patients with pre-hypertension. And these patients, remember that patients who have hypertension, the first arrhythmia is atrial fibrillation. Those patients that were treated with inhibition of the renin-angiotensin system, in this case spironolactone, developed less atrial fibrillation than those that were treated with the usual care. And this is the famous emphasis heart failure trial in patients with class 2 heart failure. As you can see, it is possible to prevent if we treat the patients early enough. This is the slide that has been presented by Professor Dobrev, and what I'm going to do is, all the information, most of the information came from his lab. Just what I'm going to do as a pharmacologist is to do this. And we have a lot of targets. Many targets. Once again, I'm old enough, and I look what I know. And look at this. I know that ibabradine, but ibabradine is an anti-anginal compound. Oh, yes, it's an anti-anginal compound. But it's a stabilizer, it's a radicals. It's a record. And carvedilol? Oh, carvedilol. I remember that I was one of the referees who, for one paper, demonstrated that carvedilol has 2 metabolites, and these metabolites are 1,000 to 10,000 more antioxidant than vitamin C. So if we look for antioxidants, we already have on the market flecainide, propafenone, ranolazine, and dantrolene. There is a very recent paper on ventricular fibrillation that they are inhibitors of the ryanodine channels. So we have a lot, as you can see, many targets, right? So the question is, can we really restore calcium handling in patients with atrial fibrillation? The answer should be yes, but I need the clinical trial to address this question. And something that we miss sometimes, and is, all the compounds are equal? No. Look, if you want to block the release of calcium from the sarcoplasmic reticulum, of all these beta blockers, only one is effective, carvedilol. If you want to see how many class 1 compounds are able to inhibit the release of calcium from the sarcoplasmic reticulum, only propafenone and flecainide. Another possibility is to modulate the number of channels that are on the surface of the membrane. And this is a paper published by Schumacher and Martens, and they demonstrate that quinidine is able to reduce the number of IQ channels. This is a selective effect because it does not happen with other channels and is stereospecific. Quinidine does it. Quinine, no. We did the same. In this case is KIR 2.1 channel density can be increased by flecainide in patients with Andersen syndrome. And it's interesting, there's no current in this patient, and you can see that when incubated in flecainide, we can induce the current that was missing. So this is my last slide. Atrial fibrillation remains the mainstay of treatment, so antiarrhythmic agents are here to stay. The rational design is based on a better understanding of the antiarrhythmic mechanism of atrial fibrillation, difference between paroxysmal and chronic atrial fibrillation, between the atrial and the ventricle, to design more selective compounds. We have multiple targets in preclinical models. We must understand the role in human atrial fibrillation. Ionic channel inhibition remains the main therapeutic strategy. It's very nice to know a lot about the new targets, but please, did you realize that after 30 years we described and we understood that flecainide and propafenone can also exert an effect on the sarcoplasmic reticulum? So we need also to understand the mechanism of action of the old drugs. Structural remodeling, the arrhythmogenic substrate and a therapeutic target. Different comorbidities means different arrhythmogenic substrate and different therapeutic strategies. Early treatment of comorbidities may reduce atrial fibrillation occurrence, and the main limitation is that we want a drug for all season. Forget it. Atrial fibrillation, treating atrial fibrillation as a single entity, this means one-size-fits-all approach is doomed to failure. Thank you.
Thank you very much. Wonderful presentation. Questions for Dr. Tamargo?
No questions.
Questions for, this was a great presentation, come on. Students, fellows, yes.
Here, and yes, we have here a lot of things about calcium and remodeling, but nothing about the role of calcium channel blockers as a prevention of atrial fibrillation recurrence. Could you summarize the evidence on this topic?
Yeah, if we look at the calcium channel, well, first of all, L-type calcium current is decreased. So to use a calcium channel blocker is not the best target. There is also evidence that sometimes with verapamil, it has been described, and with diltiazem, that sometimes they can induce atrial fibrillation. Look at the opposite. Can I overexpress L-type calcium channels? Don't do it. We worked 20 years ago with Baker 8644. All the animals die beautifully from ST elevation and marvelous vasospasms, coronary vasospasms. So we can do almost nothing with calcium channel blockers. Nevertheless, calcium channel, I belong to the age of the verapamil and I like these compounds. So please note that the current is decreased. So these compounds are not the best. They are excellent for AV nodal conduction. That's different.
Of course, thank you.
So I have a question. The ranolazine idea of sodium channel blockade, specifically in the atrium, but the idea is that it's blocking the residual sodium current. That's what some people are suggesting that
some
it's doing.
Some people suggest.
That's right.
Yeah.
So
the problem is that if it's true, while lidocaine, that's a pure sodium channel blocker, is the only antiarrhythmic agent that is not effective, amycilidine, are the two only compounds that present selective INA channel blockade and fast kinetics.
right.
And they are not effective in atrial fibrillation.
There you go. So there's another one. The sodium channel is already down-regulated, the same as the L-type calcium current. So that's another issue. Thank you very much. So the next presentation is really wrapping up all of this and bringing everything that we discussed today to the clinic. Is it a good idea to target sources of atrial fibrillation? And for that, Felipe Atienza is going to give us the results of the radar, which he really spent many moons pondering about how to do this clinical trial in Spain, and he was incredibly successful. So I'm really proud to present Felipe with his talk. Felipe.
Thank you, Pepe. Let's see. Okay, here. Thank you, Pepe, and both Nicasio and Angel for their invitation to speak right after these giants in electrophysiology. I feel really honored. Most of what I'm going to introduce you has already been commented by previous speakers, but I want to show you this slide because this is what we find in the clinical setting. When we introduce a multipolar catheter in the left atrium, we can't see almost anything. Everything is disorganized. Where can we see the order? Where are the rotors? Where are they? Let's see if we can find them. Gordon proposed this multiple wavelet hypothesis that you all already know. And in the late 90s, Heisinger showed us that some cases of atrial fibrillation were triggered or even maintained by ectopic foci that were located at the pulmonary veins. So everybody ran to isolate the veins using different strategies, segmental isolation or circumferential ablation. And what we learned from those studies was that drugs gave us around, in the best of the cases, 60% of success rate for maintenance of sinus rhythm, whereas ablation could be more effective, especially if it's supported by multiple procedures or drugs. That was, with those results in hand, both the ERA and AHA considered ablation as a class 1A indication for patients with symptomatic atrial fibrillation refractory to drugs in experienced centers. But the procedure, as you all know, is difficult, requires time. Ablation in some cases is quite extensive, and complications, although not very frequent, they can be serious. So our question 10 years ago was, could we be able to develop a technique to ablate, to treat the mechanisms, so-called pathophysiologic ablation technique. And we started with experimental data demonstrating that the left atrium that fastest sites in the left atrium were activating atrial fibrillation and were responsible for maintenance. This paper is really old, but shows that localized ablation of sites with fastest activation could terminate atrial fibrillation. This is from the 1920s by Thomas Lewis. And Dr. Halif's group showed that this slide has already been presented, but he showed that fastest sites were mostly located in the sheep heart, in the left atrium, giving rise to a left to right frequency gradient of activation. And these data were confirmed by several other investigators, demonstrating that both in persistent and paroxysmal patients, the left atrium activated fastest than the right atrium or the coronary sinus. Not only that, in experiments, such as this case, ablation at fastest activating sites, in this case in the pulmonary veins, terminated atrial fibrillation, and also in patients, this study produced by Heisiger group in collaboration with Dr. Halif's group demonstrated that almost 90% of paroxysmal cases terminated while ablating fastest activating atrial sites, most of them located in the left atrium. With this in mind, we didn't know what were the mechanisms responsible for maintenance of atrial fibrillation. Was a multiple wavelength reentry, was a localized functional reentry, so-called Rotors, or was a focal activity fastest activating the atrium. We conducted this study where we used adenosine to mimic acetylcholine function, and this activation of adenosine of the IK80 channels produce exit of the potassium, giving rise to slowing of pacemaker activity of sinus cells, and also shortening of the action potential. Dr. Halif's group demonstrated that infusion of acetylcholine accelerated rotors and increased left to right gradient, demonstrated that in this case, in these experiments, atrial fibrillation was maintained by a reentrant activity dependent on IK1. In patients, we use adenosine, easier to use than acetylcholine, and we also demonstrated that in paroxysmal atrial fibrillation, it could be accelerated, the activity of the fibrillation, both in the left and the right atrium, but specifically in the left atrium while infusing adenosine. And this can be shown in this slide where you can see that fastest sites were located in the pulmonary veins, and after adenosine infusion, those sites, the frequency of those sites significantly increased. And we aim to target those fastest activating sites in the left atrium, and we found that we were able to terminate atrial fibrillation, as shown in this slide, and isolate these fastest activating sites. With that in mind, we analyzed what happened with those patients, and in this paper we reported an observational data, only observational data, and we found that isolating the veins and ablating fastest activating sites, both in the left and the right atrium, we were able to obtain a high success rate, both in paroxysmal and not so good in persistent cases. And we also learned that in persistent patients, fastest activating sites were located not only in the veins, like in paroxysmal patients, but also in the body of the left atrium, right atrium, or the coronary sinus. So with all that in mind, with that background, in 2008, we decided to start this randomized trial called Radar AF trial, where we analyzed what happened with radiofrequency catheter ablation of drivers and compared that strategy to the standard strategy used by most of the laboratories, which is circumferential pulmonary vein isolation. The objective of the trial was to determine the efficacy and safety of this high-frequency source ablation as compared to the standard CPVI ablation in patients with indication for atrial fibrillation ablation. This was a prospective, multi-center, single-blinded, randomized one-to-one clinical trial where we used the EnsightNavX system with the specific software that enabled us to measure dominant frequency in a sequential fashion. So we move from one side to the other in the left and right atrium to measure what was the dominant frequency at those sites. We followed patients for 1 year with serial Holter recordings at 3, 6, and 12 months, and analysis was intention to treat, with a primary endpoint of freedom from atrial fibrillation at 6 months, post first ablation procedure of antiarrhythmic medications, and secondary endpoints, freedom from atrial fibrillation at 6 and 12 months, freedom from atrial fibrillation and atrial tachyarrhythmias, incidence of pre-procedural complications, adverse events, and quality of life. And we compared these strategies. In paroxysmal atrial fibrillation, we compared the standard strategy of circumferential pulmonary vein isolation with a strategy aimed at targeting high-frequency activating sites. This is an example where the left posterior view of the left atrium in purple is shown the right inferior pulmonary vein, which was isolated. We didn't target the high-frequency site inside the vein, which may cause stenosis, but we isolated that single vein which showed the highest frequency of activation. In persistent patients, since we already knew that high-frequency activating sites could be located either in the body of the left atrium or of the left atrium or the right atrium, we performed in all of them by protocol pulmonary vein isolation, but also targeted highest frequency sites located either in the left or in the right atrium. This flowchart of patients we included after considering for eligibility more than 800 patients. We randomized 232 patients, 115 paroxysmal, 117 persistent. The baseline characteristics of paroxysmal patients were basically similar. There was no significant difference between groups, the control versus the highest dominant frequency ablation group in paroxysmal patients. And the same can be said about persistent patients, were comparable. Both groups, there was no significant difference among them. Procedural characteristics. In paroxysmal patients, atrial fibrillation was induced in around 80% of the patients. Mapping time was added to the procedure in 31 minutes in paroxysmal atrial fibrillation patients. Obviously, there was no need for dominant frequency mapping in control group. Fluoroscopy time was similar. Procedural time was also similar. There was no difference. A median of 3 high-frequency sites were ablated in the HFSA group. 18 high-frequency sites could not be targeted, either due to technical limitations to access to them or concerns about safety by the operators. A mean of 2.2 pulmonary veins were isolated in the highest frequency sites ablation group, whereas a mean of 3.79 pulmonary veins were isolated in the control group. Additional lines were created in 3 patients in the control group. Mean RF time was 42 minutes in the CPVI group versus 33 minutes in the high-frequency sites ablation group. Sinus rhythm conversion was more common in the high-frequency sites ablation group, and there was no difference in the number of redo procedures between both groups. In the case of persistent patients, I remember you that all patients underwent CPVI, and in the treatment arm, we added to CPVI high-frequency sites ablation. There was no difference. Patients did not need to be induced Most of them were in atrial fibrillation at the time they arrived to the lab, but the procedure added a mean of 28 minutes in the case of the treatment group. There was no difference in fluoroscopy time or in total, but total procedure was longer. Total procedure was longer in the treatment group, in the combined group, what we call the combined group. In the combined group, 3 high-frequency sites, a medium of 3 high-frequency sites were ablated, and 26 high-frequency sites could not be targeted. Most pulmonary veins were isolated in both groups, and additional lines were created in 11 and 22 patients in the combined group. That difference was significant. There was no difference but a trend to longer radiofrequency time in the combined group, no difference in sinus rhythm conversion rate, which was very low in any case, and no difference in the number of patients undergoing reduced procedures between groups. This cartoon shows the distribution of high-frequency sites in both types of patients. As already found in other studies, in paroxysmal cases, most of high-frequency sites were located in the posterior wall, either at the PV antrum or the posterior wall, but they could also be found in some cases in the atrial appendages, the superior vena cava, or the coronary sinus. Whereas in persistent patients, the distribution of high-frequency sites was more broad, more spread around the atria, and the incidence of high-frequency sites was higher in the left atrial appendage, in the body of the left atrium, also in the pulmonary veins, and also in the right atrium. And the outcome. I didn't mention, and that is important, that in paroxysmal patients, what we aimed was to demonstrate non-inferiority. That is, able to demonstrate that burning less, only targeting high-frequency sites, was equally effective and safer than the standard protocol, that is, ablating the CPVI. Our endpoint at 6 months could not be met. In this non-inferiority analysis, we couldn't demonstrate that burning only the high-frequency sites was as efficacious as performing standard CPVI. But at 1 year, considering reduced procedures, we were able to demonstrate that the strategy of high-frequency sites ablation was non-inferior, as efficacious as the standard circumferential pulmonary vein isolation. And that was true for atrial fibrillation, freedom from atrial fibrillation at 1 year, and freedom from atrial fibrillation and atrial tachycardia at 1 year. And most importantly, we were also able to demonstrate that the incidence of peri-procedural complications and serious adverse events during follow-up was lower in patients assigned to the strategy of high-frequency sites ablation, meaning that burning less may be safer and equally efficacious than the standard CPVI strategy. The efficacy endpoints are shown, the Kaplan-Meier curves show no difference whatsoever in time to atrial fibrillation or atrial fibrillation after one procedure or after reduced procedures, and the same can be said for atrial fibrillation and atrial tachycardia after a single procedure or after multiple procedures. In the case of persistent atrial fibrillation, the study was designed to aim to demonstrate superiority in a sense that burning not only the pulmonary veins, isolating not only the pulmonary veins, but burning other sites, activating at high frequency elsewhere in the atria, wherever they were, could be better, could increase the efficacy of the procedure. But we were unable to demonstrate that. We were unable to demonstrate superiority. There was no difference at all for the primary endpoint of freedom from atrial fibrillation at 6 months, or the secondary endpoints of atrial fibrillation, atrial tachycardia at 6 months, or at 1 year. Regarding safety, peri-procedural adverse events were similar between both groups, but there was a trend a strong statistical trend for a higher incidence of complications during serious adverse events during follow-up in the combined group of patients. These Kaplan-Meier curves show that there was no difference in time to first atrial fibrillation after one procedure, although you can see that there is a trend for a better outcome in patients in the group of combined CPVI plus high-frequency site ablation, but no statistical difference whatsoever. When serious adverse events are compared in both groups, you can see, well, this is the numerical occurrence of the different adverse events, procedural adverse events, specifically tamponade or pericardial effusion were more common in the paroxysmal CPVI arm, whereas in the follow-up, atrial flutter or flutter requiring hospitalization was the main cause for serious adverse event in that group, in the CPVI group. Regarding persistent patients, as I already mentioned, adverse events, both peri-procedural and specifically adverse events after 1 month, were more common in the combined ablation group. Quality of life improved in both arms using a specific questionnaire, but we were unable to demonstrate differences between both treatment arms, the standard CPVI versus the high-frequency sites ablation group, in both paroxysmal and persistent patients. So in conclusion, high-frequency sites ablation did not reach the statistical significance for non-inferiority as compared to CPVI to achieve the primary endpoint of freedom from atrial fibrillation at 6 months, but it must be stressed that that was after a single ablation procedure. High-frequency sites ablation was non-inferior to CPVI to achieve freedom from atrial fibrillation and freedom from atrial tachyarrhythmias at 1 year, considering patients that underwent reduced procedures. And most importantly, this group had a lower incidence of severe adverse events. In comparison, in persistent patients, CPVI combined with high-frequency sites ablation offer no incremental value, did not prove to be superior, and even saw a trend for an increase in complications risk. And we consider that these results may offer a novel mechanistic treatment paradigm for patients with paroxysmal atrial fibrillation. This is the end of the session, and I want to end with this study that was really a translational study that started in 2004 with that adenosine paper that I showed you, and we were able to translate the findings in dominant frequency, the usefulness of dominant frequency to map fastest atrial activation and to target those sites to treat a real problem, which is atrial fibrillation. This trial and these studies were triggered by a grant by the CENIC in 2007, and were with the support, the very strong and committed support of St. Jude Medical. We also received grants from the Spanish Society of Cardiology and the Network of Investigation in Cardiovascular Research. And the participants in some of these studies are listed here, and most of them have been mentioned already. And the most important people are the investigators who were committed, they contributed, and they worked really hard. I must say that this trial was really hard to recruit patients, to perform, and the work was superb. And above all, I want to thank Professor Halife for his permanent support and mentorship. I thank you for your attention.
Thank you, Felipe. While you think about your questions for Felipe, I'm going to ask the speakers to come to the podium. We're going to spend 15 minutes in the general discussion. I know the time is late, but I think some people may have burning questions. I'm sure the fellows and the students are going to be jumping to the microphone to ask questions. So can I have Dobry, please, Juan, Sandeep, Ricardo, if you want to sit there. Any questions for Felipe? Okay, I have a question for you. So, the radar was successful in demonstrating non-inferiority for paroxysmal atrial fibrillation for the dominant frequency side ablation versus, please come sit down in the podium. And it was not successful with persistent atrial fibrillation. And the likelihood of the lack of success is probably because you were doing more damage in the persistent atrial fibrillation patients. Do you think that is there hope for persistent atrial fibrillation patients to do dominant frequency mapping or combining it with some other approach?
Well, that's the question. That's the difficult question. Certainly, not only dominant frequency mapping is enough. We need something else. And I truly believe after what we heard is that the combination of phase mapping with dominant frequency and probably combined with both intracardiac and panoramic mapping will give us the answer. But after listening to all of you, persistent atrial fibrillation is another disease, so to say, and it's very difficult, and we'll know much less from persistent atrial fibrillation than from paroxysmal atrial fibrillation. But if I had to choose, and I'm choosing already, the way we're going is to combine those tools that I already mentioned, dominant frequency with phase mapping, both intracardiac and body surface mapping.
Okay, thank you. Have a seat. I'd like to start the discussion, and I would like the audience to participate in this discussion. We have had some really wonderful presentations that go from basically from the gene all the way to the patient. And we had some really outstanding speakers, and I would like the younger guys to take advantage of that. This is usually a unique opportunity. So I want Julian to ask the first question so that he would start the discussion.
Like a fellow.
Like a fellow.
Okay, congratulations to all the speakers, but Juan, I got depressed after your talk. I got depressed because, and I can invite you to perform ablations in our center because it's going better, the ablation than the drugs. But no, talking seriously, what about the other face of the coin, the other side of the coin? How to improve the tolerance and to avoid secondary effects of the drugs with phenotype, genotype, any kind of improvement?
Yeah, I think from this point of view, I didn't touch this point. I didn't touch genetics because, but it's quite clear at the present time that, for instance, in Spain, probably some of you must ask for quinidine for some of your patients because quinidine has disappeared. And we know that there are for some patients, there's a couple of patients, a couple of papers from Eleanor and from people from Denmark, that there are some patients that we identify, some of the mutations, probably they can have a specific treatment. There are 2 or 3 very nice examples. One is quinidine in some patients with Brugada, some patients with, or of course, beta blockers on the catecholaminergic. The sample of LongQT3 with ranolazine, the paper published by Arthur Moss a few years ago in 6 patients, with a QTc over 650 milliseconds. So from this point of view, antiarrhythmic agents, this is something that I didn't touch here. I just want to mention that it's not to feel depressed. I mean, as a pharmacologist, probably I feel feel fine because there are a lot of targets. We have targets. The problem is we need to validate the targets in humans. That's the point. But the problem is if I go to calcium handling and I said I don't have drugs. If I go to different channels, I don't have drugs. We have drugs. We have many drugs. The point is that we are in the basic level. And I want to stress that people don't like to give the next step, and is put your money and go to phase 2 trial. Go to a phase 2. And most of the phase 2, with the exception of Remodeling, are proof of concept. Well, I'm generous. I call them proof of concept. 20 patients with atrial fibrillation as a phase 2 means nothing for me, you know. But this is the real problem. People, and also, if I look at atrial fibrillation and you give flecainide, if you give flecainide to the people where it is indicated, there are 2 meta-analyses that they didn't found that you increase morbidity or mortality. The problem is that your worst patients are not suitable for flecainide. So we learn that we must learn who, to whom, how to handle or how to prescribe antiarrhythmic drugs. But how to improve them, safety.
One of the problems that you will have is convincing the pharmaceutical
Yeah, that's the point.
companies.
That was my point now. I mean, if you go, I underline some of the drugs just to tell you that they look at this slide and they say, oh my God, these compounds have already been commercialized, like ranolazine or carvedilol. They have a price. The price will go down when I go into the market. I'm not going to put my money in this field. And if you look how many of these companies are interested, you can correct me, but probably ranolazine, AstraZeneca because of tradition, but this morning I listened. I heard the newspaper on financial affairs, and AstraZeneca is ready to be picked up by Pfizer. So I don't know if they will continue with this field because Pfizer has his own compound and is not very happy. So this is the problem. The pharmaceutical companies don't want to invest money in this area. Okay. Not at all.
So let me continue and switch gears a little bit. Jesus Almendral has a question there, but before Jesus asks his question, I want Omer to. I know I interrupted him because time was getting long, but. Omer, one of the important messages that I got from your presentation is that the body surface mapping is a very good idea, but you have to be very careful. Some people now are commercializing this body surface mapping. What's your? Give me in just a couple of words what advice you would give to people who are doing body surface mapping. You think they should take it to the EP laboratory and do ablations based on body surface mapping as we know it today? Just very briefly, please.
Well, I hate to be my kids call it the party pooper, because
I want you to be controversial, that's what I want.
Yes, so that's I am. I think I'm creating myself a name of a critical person, because what happens with these 2 systems, I will expand a little bit on the basket from Narayan and the body surface mapping approach. I think they have been, they are a little bit oversold, a little bit, there is a lot of excitement, of course, because of the findings. However, we have to be very careful of interpreting the data. And actually, in our lab, we are stepping back a little bit, trying to validate those approaches. I believe there is a lot of data that we could extract from the atria from body surface, using body surface mapping and the panoramic mapping from the inside. I think there is a lot of to do with signal processing and considering those, the frequency domain, the phase domain, and others that I don't have time to expand on. I think the commercialization is a sign of the time, I guess. That people are rushing to commercialize things. I think we are in trouble because we are, in a way, in the hands of the industry, and that's true for the pharmaceuticals as well as the device companies.
I wanted you to be controversial, but not so controversial. So.
Okay. I'm not a good politician. But I think, yes, we are in trouble. I think an understanding of what work needs to be done. For example, you mentioned that I may switch a little bit from devices to pharmaceuticals. I guess the key here is in understanding this individual targets in each patient, and that may not be even genetics, it may be mapping the substrate in situ, in vivo, to know what are the particular targets of each patient. It's probably a very expensive endeavor, and it's probably that these are questions that should be addressed outside of this room. But indeed, there is an infinite effort that you should put to.
That's a good point. That's a very good point.
Solve a problem.
Thank you.
Yeah, I'm in the same boat as everybody.
Good. Jesus, please.
My question is for Omer Bernfeld. And the question is, the problem I have on, it's for you, how to annotate a signal in atrial fibrillation. I mean, coming from clinical electrophysiology of tachycardias, what I learned is that the strength of the intracardiac signal is that it tells us when the activation crosses the site of where the catheter is located. Good. Then with VT, I learned that the signal could be fractionated, meaning slow conduction. But now, during atrial fibrillation, I am puzzled anytime I see, you have shown here several signals, or if I had to annotate, I would say, I don't know, it could be fractionation, slow conduction, 1 beat, or it could be several activations of the same site. And Pepe insisted, and it's very important to know the rate, because that is the determinant of a lot of things. So do you think the way to go is to be able to better annotate signals, or to consider the signal as a unit and consider the signal together?
Let me start with putting in question your first assumption that the electrode, the deflection in the electrode shows when the action potential crosses the vicinity of that electrode. I think that perception is already inaccurate in several times. It doesn't consider far field contribution, which we cannot remove at any point. And I would like to emphasize that neither unipolar, of course, as everybody knows, but also bipolar, cannot tell where the signal is coming from. And that's a physical reality. I mean, we may have experience, we ablated things, we saw many cases, and we have the intuition that action potential is getting close or far away from us, it's crossing at this particular moment our electrode. But that's maybe not true all the times. And in that case, we're in trouble because electrical recordings suffer from that limitation. I call it, in physics, we call it non-uniqueness. There is infinite possible situations that can give rise to a single type of electrogram. And we've discussed that with Felipe Atienza many times. We realize indeed that we have to map using body surface mapping, the intracardiac recordings. There is no easy answer for that. I think optical mapping also suffers to some extent from that, but I think that may be a better situation. So my approach is to take with a grain of salt interpretation of any signal, and that includes dominant frequency. I don't think that single parameter can describe so many things that happen in the atria during fibrillation, certainly.
This is a discussion that has been going on also between Sanjeev Narayan and Maurits Alessi. Maurits Alessi claims that the filtering with the dominant frequency mapping is really an artifact, and that's why Sanjeev Narayan sees rotors.
Yeah, well, I don't think it's an artifact. Okay, well, it's an artificial, it's a processing, yes, but it's something that is physiologically present. I think it's significant in terms, it's not just number crunching that gives you numbers that do not mean anything. There is physiological meaning to that. So, of course, that I've been saying that to Maurits and Uli Schotten every time we have a discussion. It's something physiological, and the filtering, indeed, it's affecting our signal. I think we have to understand what is it doing to the signal, and that was my effort in the last part of my talk. What is that high dominant frequency filtering doing to the signals? And I showed in simulations, and of course the burden is on me, of course, to show that more convincingly maybe, that this is indeed a physiological entity. It's not just number crunching. I believe that there are more important or less important features in each signals. It, of course, depends on the question. If we are looking for drivers, a signal may have signature that is less or more important to that, to that driver. And it's important not only ablation, of course, but to understand mechanisms that will lead us to new targets or better targets, pharmaceutical targets.
Okay.
So I think the mapping is essential, and it's an ongoing research right now.
Thank you. So while you think about another question, I'm not going to ask all the questions. I'm going to. Okay, good. Let me just ask my question, and then I'll get to you guys. Dobry. The work that you're doing with the patients is the most difficult of all the work that is done, because you have accumulated all these data over years that you obtain patient information, and the myocytes that you isolate come from patients which are not homogeneous necessarily. Is that the case? Am I interpreting this correctly? And if they are not homogeneous, how do you interpret those results to make sure that you're getting meaningful data?
This is a big problem, and the problem is that the cardiosurgeons, I apologize for this, they don't document that much. You know, there is not echo for all the patients. You have no real numbers frequently, how big is the left atrium, the right atrium. It is very, very few information. What we have is, you know, we have indications, we know age, we know whether there was a coronary artery disease, a bigraft basic or wave replacement. You know, we know probably something about the arrhythmia, we find some ECGs, but it is not that much. I'm trying now in the new place to have a much better, you know, documentation, but it's a big problem. We are looking at the left ventricular ejection fraction. It is, of course, how is that measured is an issue. The second one is we can just tell you that we don't have in the average, major differences in systolic dysfunction. We have no idea about diastolic heart failure. It could be what you're saying. It could be that for whatever reasons, patients with paroxysmal atrial fibrillation, remember, our inclusion criterion is open heart surgery using the lung machine. So it is a very, very selected population. And it could be that for whatever reasons, these patients with paroxysmal atrial fibrillation have much more frequently diastolic heart failure. I don't want to go into the definition of this, because it's everything what is not systolic is in fact diastolic heart failure, and all the epidemiologic data now suggest that about 50% of patients with heart failure are probably having exclusively diastolic. It could be that this is the reason why we see this behavior in DADs and so on. But I was surprised to see that this behavior at the cellular level was much more easier to study in paroxysmal AF versus chronic persistent atrial fibrillation. And one reason could be that the high atrial rate probably silenced the calcium signaling changes. We have a paper with another group in an animal model where you see that the very high atrial rate rather reduces, as you see in your model, expression of ryanodine receptor channels, decreases phosphorylation. This is okay, but you know, it is not in the context of the comorbidities, it's not in the human. There's a lot of factors, and I really believe that these data are true, but I also believe that our data in humans, we don't know to justify the model because we have the diagnosis. You know, you can discuss whether this paroxysmal AF patient is the same that others. They for sure not. Yeah. And I was surprised to see this behavior, which is pretty consistent with this in our hands. But remember, we spent 6 years to produce this data for the last part of my talk, collecting patients, you know, again and again. You know, the isolation procedure is difficult. So there's a lot of stuff what
Absolutely.
you can consider.
Actually, I was pleasantly surprised by the data showing that the action potential durations were not different, indicating that in those patients remodeling was just early remodeling that was reversible.
I can tell you that what we did in these experiments is we isolated myocyte without calcium buffers. You know, traditionally for patch clamp experiments, we have a lot of calcium buffers to protect the myocyte during the procedure from calcium-induced cytotoxicity. If you do that, you cannot measure calcium transient and calcium signals later on. And we measured it. So our procedure for these 2 papers is completely different from all the rest we published the last 15 years. And I'm sure that most of the papers, except this with so-called perforated patch, they have artificial compositions and so on. So our data are not, we didn't see the differences. We have another project where we use rather calcium buffered conditions with some tendency to see the reduction because we buffer something which offset the differences. You know, this is not physiological. You can, of course, certainly under certain conditions measure shorter action potentials, but I personally now believe they are not real.
Okay. And who has a microphone there?
Can I make a comment on this point?
Can you wait for a second? We had some
Yeah, okay.
people here. Can you pass the microphone here?
Can I make a comment, Pepe?
Yes. Who are you?
Me. Okay.
Sorry,
it's
I'll
just
get back to you.
what
Okay.
Professor Dobrev already mentioned. This paper takes 6 years. It's very easy. Why we don't join together for translational medicine and for translational blah blah blah. And then you have somebody who is going to a hospital one day and another day, another day, another day. Yeah, every day. Yeah, to get tissue, and sometimes you get 1 or 2 preparations, and for some of them. Sometimes you take, I remember, the left atria is 1 every, and because we have friends, and I don't want to point anybody, but it takes, the referee answers you need 3 more left atria. You say, my God, I need 6 months if I'm lucky enough.
Okay, thank you very much. So, we need to move on. You have a question?
Yes, I have a question for Dr. Dobrev. And it's interesting that the calcium transients may induce or may initiate atrial fibrillation, but it's also, and it makes sense. And my question is, do you have any data on the behavior of those calcium transients from young patients and old patients? Do they behave different when they are in sinusoidal or they are in paroxysmal AF? And the second question, and probably you had data about that, is what happened when you use isoproterenol on those calcium transients? Also from sinusoidal patients and paroxysmal AF patients.
I can tell you about age, we cannot say that much because all the patients are old. What we have, they have around 70, 68, 70 age in sinus rhythm. Chronic persistent atrial fibrillation is the same. This is paroxysmal. This is just because I can repeat it again and again, because excess is open heart surgery. We are not asking for what the patient is. We just can get tissue. They are older. They have, this is as we don't know. in chronic persistent atrial fibrillation, we systematically used isoproterenol as rather than supplemental files, and no one realizes because the people never look in supplemental files. And we have 18 figures in our circulation paper, 18 supplemental files. We can probably, smaller journals have 3 papers just with this data. There is no difference. The difference is before.
We have 17.
Yeah, the difference is before, but the response to isoproterenol, calcium transients, L-type calcium current, all that stuff was similar as in patients without atrial fibrillation. But the calcium transients are different. They are reduced by 50% in patients with long-standing persistent atrial fibrillation. There is no change in paroxysmal atrial fibrillation. So it's a big difference in the properties between the two populations.
Please, ask your question.
Okay, just an EP lab question, about a very fellow question about what you have. What you have presented previously about the slice of the posterior wall of the left atrium and of the pulmonary veins. When it's got more fibrotic, you have shown the rotors get organized and kind of flatter. Yeah. It's true, but there is one figure I get the question about is that the small spot of fibrosis where it gets around just in the upper part of the tissue. How will you differentiate it from a rotor, from a microentry?
Unless you develop MRI of the resolution of less than 1 millimeter, it's going to be very hard. So you can think about anchoring to fibrosis or simply the parameters of the excitation process make the rotor very stable. You can't have it. See, the rotors depend, the relationship between excitability and refractoriness is a very, very tight relationship that if you modify them slightly, you can change a rotor from a very stable rotor to a meandering rotor. And if you modify them more, you can make it a rotor that drifts and goes away. So it all depends on the properties of the patient itself and the substrate. But if you have an anchoring site of less than 1 millimeter or so, it's going to be very hard to distinguish. You have a question back there, yes.
Barcelona. It was a really outstanding speech for all the speakers, but I wanted to focus on Felipe, a question for Felipe. When you ablate the pulmonary veins, you are completely sure that you have ablated the veins because you have a catheter inside the vein and you see that it's isolated. But which is the target when you face the high-frequency ablation sites? Which is the target really? Because I see that in the persistent AF group, you have a very low conversion to sinus rhythm. Which is the target? Just to slow the frequency? How can you be sure that you have completely resolved the problem of the rotors?
Yes, it's a very pertinent question. The target was burning at least 4 high-frequency sites in the left atrium until AF converted to sinus rhythm, or 3 sites in the right atrium or the coronary sinus. If it didn't convert, you stopped. So the endpoint of the ablation procedure was sinus rhythm conversion until you burn 4 sites in the high-frequency sites in the left or 3 in the right and the coronary sinus. That was the endpoint. Sinus rhythm conversion was around 45% in the high-frequency sites ablation arm. As you say, it's not high. It was higher in our previous study, but that's what we found.
Okay.
A couple of weeks ago, in Cotignol, in Papone's lab, and he was focusing on rotors, and he's mapping the high-frequency ablation sites, and he's able to convert in every single patient from atrial fibrillation to sinus rhythm.
Who was that?
Papone. No.
Well, let me tell.
I mean, but I've seen it.
You've seen it?
Yeah,
Okay.
I've seen it. Yeah.
Then I believe you.
Yeah. Well, it's incredible to see, but he's very quick mapping, and he's converting the patients. So the question that raised here is, we are doing quite well, or we are so fast, because sometimes the rotors are stable, but sometimes they are very erratic, and perhaps we are not able to go so fast as to move from one to another.
I think we need to study the patient much more than we are right now. I think with low resolution mapping, it's very difficult to find the sites of high frequency. You really have to spend a lot of time, and the more resolution you have, the better off you will be identifying the sources. But right now, we are still far away from being able to do it. I have a question for Ricardo. Ricardo, I know that you talked to Juan, and I know that you may agree or disagree with him. So which is your favorite target?
My favorite target?
Your favorite target, is it IKS or is it IK1?
IKS and IK1, I probably say both of them, because both of them have very important features. One, for IKS is the frequency-dependent activation and the sensibility to the beta-adrenergic stimulation. And the IK1 is perfect because it's playing a role both in the control of the resting membrane potential and in the final repolarization, and as you mentioned, in the dynamics of the rotor. So IK1 is very important. But the thing is that it's very difficult to get selective drugs for these currents. But probably we should be critical with this point of view of extreme selectivity for one target, because as probably the audience would agree with me, is that probably the best antiarrhythmic drug they use is amiodarone. That is the contrary of a selective drug, is targeting many things. Right. Even amiodarone is a drug that has been demonstrated that is somewhat effective for preventing atrial remodeling. So I don't know. We are searching for selective drugs, and we probably would have amiodarone in mind. So.
Right. But I want to create some debate with Sandeep, because Sandeep does favor IK1, I'm sure, but he also mentioned that IKS is different in terms of the rotors. So I want the two of you to tell me who's right.
No, I think Pepe is right in the sense when we have overexpressed IKS, as you see in chronic AF, to our surprise, we did not find that the rotors were accelerated. Actually, we found more wave breaks and more fibrillatory.
Talk to the microphone, Sandeep.
Yeah. Mm-hmm. So I'm not sure. I mean, if it probably will definitely affect the number of wave breaks. But I think we will probably have, of course, our results were in neonatal ventricular monolayer. So I think we will have to test in an animal model. I think if IKS, I think one other problem is, in isolated atrial cells, IKS is very hard to find. So I think we'll have to probably see, say in sheep, first of all, we'll have to demonstrate if IKS is upregulated, as you see in your patients. And then to find a selective drug to see if it stops AF. So I think that would be the...
Let me introduce another debate that is worse for us, for the basic sciences in this field. That is, we have only a small piece of the atrium that is in the appendage, and we see heterogeneity in this small piece, but we don't know what happened at this level in the whole atria. Probably things are much more complex than we know now. So,
absolutely. Thank you. You have a question? Yes.
It's a question for Dr. Atienza. I would like to know if in the group of patients of dominant frequency group, in REDO patients, you perform again a dominant frequency map, and what happened?
Yeah, you mean the redo procedures in the high-frequency sites ablation group. What we found was very interesting, I didn't mention on that. It was that the most common cause of recurrence was reconnection of the highest frequency sites that were previously isolated. Meaning that I think that the main outcome of our trial is that the PVs are critical in 95% of paroxysmal cases. And the problem that we have with atrial fibrillation is, paroxysmal atrial fibrillation, is that we are unable to obtain persistent PV isolation. So as you see in your clinical experience, when your paroxysmal cases recur, most of the recurrences are due to reconnection. And that's what we found. But in the case of the highest frequency sites ablation, what we found was that we only needed to ablate the highest sites, highest frequency sites, and not ablating the rest of the veins was what provided safety to the trial. So that was an answering to what Julio was saying. Our aim was to be efficacious but also safe. Safety was important for us, so we are aware that maybe we miss some sites, but it's more important for us, it was more important for the trial to avoid harm than to obtain a high rate. And besides, I do not believe anybody that tells me that he's able to do whatever 100%, whatever he talks about, either sinus rhythm conversion or whatever he's talking about. 100% is always lie.
Okay, any other questions? Yes, back there, please.
Yeah, I'm Guillermo Perez here from Girona. Speaking about targets, I have a question for Dr. Tamargo. In one of your slides, you mentioned BK channels. Is that accidental, or do you have any comment on that?
Yeah, I get the data. Well, they already published one paper on the topic, so at my right side very recently. It's a very interesting new target. As usually, you have both sides of the coin. In my slide, I present what happened when you knock the current. When you knock the current, there's a paper published in Journal of Physiology by Lee. There's a significant prolongation of the action potential duration. But this prolongation of the action potential duration sometimes was followed early after depolarization and induction of AF. There is also a paper published in Heart Rhythm 1.5 years ago indicating that to block slow conductance can induce proarrhythmic effects. But at the same time, no doubt, if you want to prolong the action potential duration in the atria, this is a very nice target. So once again, we have both sides of the coin, right? And I just skipped the slide. I did it. I mean, I was tempted to stop for a moment. There's another point, and is we must define the role of this target, of these channels, of the different isoforms in the atria and the ventricle. Second, be very careful, because the expression is significantly changed if you have not only atrial fibrillation but also heart failure or other cardiac disease. And the third point is we need to develop selective compounds against the cardiac one, because you have data, and I think that you have data on vascular smooth muscle cell, Guillermo, of the old times, and you know that you can get funny results in the vascular system.
Okay, well, I think it's time. We went over for half an hour, but I think we had a good time. I hope the audience had a good time. I hope that this group of presentations will be helpful for you in the future. Perhaps right now you just see filaments moving from one part of your chest to the other part of your chest. But I think this information eventually is going to help you in your practice. I want to thank the organizers, the Sociedad Española de Cardiología, and the Arrhythmia Group for allowing us to present such basic information, but at the same time for letting us have a good time. I think this group of presentations has been very unique from my own perspective, because you rarely see at meetings, at conferences such as this one, presentations that go from the very, very basic all the way to the clinic, and that they can coordinate themselves and they can sort of mingle together. So I'm very proud of this talk. Nicasio, thank you very, very much. Angel, thank you very, very much. It's been wonderful. Thank you, guys. You did a fantastic job. You're great, all of you. I'm proud of you. And I want to thank the audience, and good night.